welcome all speakers of today, Professor Gauri Saxena, Professor Rita Kundu, Professor Nirmal Banerjee in this session, plenary lectures and keynote lecture. So I request our host to start this. Okay, and Naranda, you please chair this. So our host will introduce everything. Thank you very much.
Thank you, ma'am. A very good morning to all dignitaries, delegates, participants and guests. Myself, Devasmita Mukherjee.
I am Kunal Kumar Shaha.
We are very fortunate to be your host, and it's our privilege to welcome you all in the second day of today's national seminar on cytogenetics in vitro culture and phytochemistry of plants and microbes for sustainable use.
I am taking this privilege to briefly announce today's schedule, which include one keynote lecture, two plenary lectures. Following this, there will be the oral and poster presentation. Then we will have a lunch break. And following that, we will have finally the valedictory session. Now, moving towards the main session, I would like to request Professor Narayan Chandra Mondol, Senior Professor of the Department of Botany, Visva-Bharati, to chair this keynote lecture session. Sir, please.
Okay, thank you, Kunal and Devasmita, and thank you, Angelika, Muthimita, and all the organizers, HOD of this department, for giving me this chance to chair this session. So very good morning to all of you. It is a very rainy and basically gloomy type of weather today in Santiniketan. Although it is supposed to be a very bright sunny day this time during the month of January, this 23rd of January. Today is the birth of Netaji Subhash Chandra Bosu. So we salute our national hero. With this, I welcome all the audience, and although we are not getting bright sunlight, still I hope that we'll get a fragrance of rose from Lucknow, Nawabi Gharana ka fragrance of rose through geranium, and Madam Gauri Saxena will give this flavor to all of us. So I welcome Professor Gauri Saxena from the University of Lucknow and CSIR CMAP. And regarding today's speaker, Professor Gauri Saxena, Madam, she did her PhD from University of Lucknow, CSIR CMAP. And she has completed her early education, undergraduate BSc Botany Honors from University of Delhi, MSc from University of Delhi, and later she has completed her PhD under the supervision of Professor Sushil Kumar, renowned scientist, renowned botanist, under the supervision of Professor Sushil Kumar in the University of Lucknow and CSIR CMAP in 2001. And before the completion of her PhD, she had joined in service in the 1996, and since then she has from assistant professor to associate professor and professor in 2011. And she is a renowned biotechnologist in our country, and because of her work, because of her contribution in the biotechnological work in tissue culture, and she has awarded one for her work, and these are the major things. And regarding her work, about 26 years in the field of teaching and teaching postgraduate and undergraduate at the Department of Botany, Lucknow University, and research at CMAP, Central Institute of Medicinal and Aromatic Plants, Lucknow, CSIR, India, and Department of Botany, Lucknow University, Lucknow. The initial work was carried out in the Genetic Resource of Biotechnology Division of CMAP, CSIR. It involved tissue culture and genetic transformation techniques. During this period, useful soma clones of rose-scented geranium, Pelargonium graveolens, where the scientific name, I do not know that I will be happy if Madam shows this plant somehow. So the common geranium, okay, I do not debate on it, and this is my ignorance. So when I'll see, I will be all like me, others will be happy. Being a botanist, I should not utter these where rose-scented geranium were generated and stably established and propagated for several generations under stringent field conditions. Genetic transformation studies were carried out using super virulent strains of Agrobacterium rhizogens Several transformants were also produced in Pelargonium gravulans and Pogostemon pachauli, of which few showed improvement over the existing cultivars in terms of agronomic characters and their essential oil yielding qualities. Besides, alternaria alternata toxin tolerant callus lines were produced from which disease-free plants were generated. The variants produced in various experiments were analyzed at molecular level too. A geranium plant with improved traits has been accepted as a US patent. This is a very marvelous thing and novel thing, and which we must happily share with my audience. So other works were conducted in the area of biochemical and molecular profiling of medicinally and aromatically important plants like Ocimum, Salvia, Pogostemon, and conifers like Pinus and Cephalotaxus, and medicinal plants like Selenium. Research works in the area of morphotaxonomy in few conifers has also been attempted by Madam. It first includes manipulation of plant metabolites using plant callus tissue culture. Attempts have also been made to establish plant tissue culture for the propagation of therapeutically important drugs and vaccines. The second objective revolves around the biochemical as well as molecular characterization of economically important gymnosperms of Cephalotaxus, Taxus, and Pinus. So very important medicinal plants. And apart from these academic positions and research work, Madam is supervising a number of 5 postdoctoral students under her in laboratory, and already 9 PhD students has been awarded by her able supervision, and 8 students are working at present. She has published 31 peer-reviewed research articles in reputed journals and 1 US patent, and several book chapters. And apart from that, Madam is involved. This is, I am particularly because I am also additionally involved in Visva-Bharati. So Madam is similarly involved in a lot of additional charges in her university. So assistant provost, additional proctor of the university, president ladies club, assistant coordinator, MSc course, Department of Botany, Lucknow University. In charge discipline committee. Disciplinary committee is a very important committee. So any kind of punishment or any kind of so students who are not doing properly, so many bad decisions or many unpleasant decisions Madam will have to take during this time. And in charge Mission Shakti, Department of Botany, Lucknow University. So these are her major works she is engaged, and a lot of research projects she is also conducting, and her label is reflected in the US patent and her research work. So with this, today she will present, just missed the title. So can you share?
Plant tissue culture in essential oil yielding plants.
Plant tissue culture in essential oil yielding plants. So Professor Gauri Saxena, Madam, will deliver a lecture on plant tissue culture and essential oil yielding plants in today's keynote seminar. So with these words, I request Madam to kindly take the dais and deliver your lecture. Please, Madam.
Thank you so much, Professor Mandal. Thank you, and thank you for all the organizing team for making me, giving me this opportunity on this platform, especially Professor Biswas, and I think there are a couple of people from the college also, the Raidighi college. And of course, Angelica, since we have been together in CMAF for a couple of years, that is how I know her. My lecture would
primarily, anyway, thank you, sir, for the kind words, but I am just very, very small this thing in the research community, because ours is a state university, so we need to, really have a balance between the teaching and the research grants that we get, because we do not get grants like central universities, so we have to work in those crunch conditions, but still we try our level best to keep up with the coming up research, whatever information that best we can gather. Yes, sir, thank you so much for the kind words. But today's talk is going to be on plant tissue culture in the area of essential oil leading plants. Why I chose this was because I have been working on medicinal aromatic plants for quite some time. And as sir pointed out, that rose-scented geranium, all right, so that is a plant. It's geraniums are of two kinds. They are one which are ornamentals and the others which are essential oil yielding plants. And the ones which are essential oil yielding plants, they generally have an essential oil which is very, very close to the rose oil. In fact, it has been used as an adulterant in the rose oil, and the composition is almost similar to the rose oil. So on elaborating on that, sir, it's called a rose-scented geranium, and the botanical name for that is Pelargonium gravulans. So that is, and belongs to Geraniaceae, of course. So that is how, and it's a high altitude plant, does not grow in plains. So we had a challenging point of getting it onto the plains, because Gangetic plains are pretty hot, and during summers, there are extremes of summers and extremes of winter. So that was pretty challenging. And my guide wanted this plant to grow in Lucknow because he was very fond of this plant. So that is how we started to work, and we had a DBT project. 5-6 years, the DBT gave us extension also, because the kind of work that was put into this project, and we were very, very successful in cultivating Pelargonium in Lucknow, and that is how it's still being cultivated, and the technology is being transferred from CMAP to the common farmers also in the plains, which was quite an achievement, because a high altitude plant being brought to plains, and then of course yielding good quality of essential oil, and the essential oil is pretty expensive in case of Pelargonium. So that is the background of whatever I did, a little bit whatever you spoke of. So I basically worked on essential oil yielding plants, most of them. Patchouli is one of them, Pogostemon was one of them, and many more, sir. And then I switched, as you see my CV, so when I came to the university, I was given gymnosperms to teach, and was given a very little part of biotechnology, because I was pretty young when I joined, and then they gave me all these lower branches to teach. So I started to work on gymnosperms. I thought, let me just explore the medicinal and aromatic qualities of gymnosperms. That is how I started to work on cephalotaxis and taxus. And we've also worked on Pinus, sir, and we still continue the legacy of gymnosperms. So I keep doing a little bit of work on gymnosperms. They're pretty intriguing plants. And believe me, they're as important as angiosperms, because most of the time we only talk about angiosperms. But since I've started to teach gymnosperms, I feel they are more, they have more potential somehow, but though they are very difficult to work on, because most of them are tree-like species, and most of them have a lot of tannins and polyphenols and so on. So therefore, gymnosperms are very difficult to work on, but they are as equally important, I feel, as angiosperms are. Presently, we're working on two very important members. This is Selenum, sir, and I don't know if you have heard of it, but that's an alternative source of Jatamansi that's being used in the tribal areas of Uttarakhand and all. And it also grows in parts of Nepal. Selenum is very, very common in Nepal. And there is one which is working on black ginger. So I'm trying to work on essential oils of them also. So primarily working on phytochemistry of plants. And therefore my talk today on the use of plant tissue culture in essential oiling plants, is going to be focusing on that. And the talk is going to be very, very basic, because I feel the basics I might be, I didn't know what kind of audience I'll be addressing, but I think I'll be addressing a lot of students. I don't see them here, but as I perceive, probably they are on YouTube. It's being, live on YouTube. So therefore, it's going to be a basic talk on how plant tissue culture has been helping in essential oil production of essentially linked plants, and what all can we explore in this area, So just, so may I just begin my May I begin the lecture, sir?
Okay, sure.
Thank you so much.
Most of our audience are they are researcher students. So you please go ahead.
Yeah, so it's going to be a basic talk like a teacher, you know, because I'm more of a teacher than of a researcher. So I think I should be able to do justice to the talk. Thank you so much, sir. So. So is it?
Yes, it is visible, clearly visible.
Absolutely. Thank you so much, sir. So the topic for the morning talk today is plant tissue culture and essential oil leading plants. And just for the basics, because most of us know what plant tissue culture is, just to go around, you know, telling that once again, just a quick review of what plant tissue culture is. So plant tissue culture, of course, involves culturing or growing of, you know, small tissues or cells, or even isolated organs sometimes, you know, using supplements in the form of growth regulators in the aseptic condition. Aseptic condition is a very important one, because if it is not aseptic, and it doesn't work as well. And of course, then you grow it artificially in the laboratory, and you try to regenerate a complete plant out of a small tissue. The idea is that each cell in case of plants is a totipotent cell and is capable of giving rise to a complete plant, all right. And tissue culture primarily, you know, has been attempted to obtain disease-free plants for germplasm conservation, for somatic hybridization, and more importantly, for genetic improvement of plants, you know, in culture conditions. Obviously, for research purposes, as you know, obtaining haploid plants and so on. And we also know who, you know, he was the father. Haberland was the father of plant tissue culture, and how he, in India, Professor Maheshwari and Professor Guha made a remarkable contribution to this field of tissue culture in India. In fact, I have been DU students, so plant tissue culture has been one of the very important prime areas of the Delhi University Department of Botany. And methods which I have generally followed in plant tissue cultures are one is your micropropagation. That is, most of people harp on plant tissue culture as, you know, as a way to achieve micropropagation. The other, of course, is organogenesis. So you can form several callus cultures and so on. And then third is your somatic embryogenesis, that you directly attempt forming a somatic embryo out of a plant tissue culture procedure. All right. The major stages involved in micropropagation has been derived by Murashige, which was one of the discovery on whom one of the medium, Murashige and Skoog's medium, has been named. So there is one, you have an explant, and you generally put the explant on an artificial media, and then you tend to grow it in presence of plant hormones. And then, of course, you have a complete plant, and that plant can be then acclimatized in the field conditions, you know, first in the glass house and then finally transferred to field conditions. And then you have a complete plant born out of tissue culture. That is how micropropagation is achieved in case of tissue culture. And then you have organogenesis, all right. So the organogenesis is achieved, of course, when you have individual organs, you know, or directly from an explant or from the callus, and cell culture is induced from these explants, all right. So you have a couple of hormonal combinations which are tried, and these hormones, which are auxins, cytokinins, and gibberellins primarily, are being used, you know, for the organogenesis. And finally, the idea is to have a complete plant. And okay, and one very important achievement of plant tissue culture has been to have direct embryogenesis from these cells or tissues or part of plants, you know. And these somatic embryos, as they are called as, you know, can be grown into a complete plantlet, all right, and they actually try to avert the middle stages of the, and or the I should be saying they surpass the stages of tissue culture, and can be forming complete plants, in very short time. We had been able to achieve, in Pelagonia, we had a cytosomatic embryogenesis directly from the hairy root, which was quite a achievement, because we could make transformants directly from the hairy root instead of hairy roots being, cultured and then regeneration being attempted in hairy roots. I'll come to the details about it. A quick glance on it. So we all know the applications of plant tissue culture, So that is basically the germplasm conservation. You have disease-free plants. You can do micropropagation. You can have genetic transformation out of it. You can even have early flowering and varieties which are tolerant to salinity, drought, heat, or stress. And most importantly, if you see, you also tend to have what you call as secondary metabolite production through plant tissue culture. Now, secondary metabolite through plant tissue culture, what you also call as metabolomics now, the metabolomics is being attempted in case of, plant tissue culture. And secondary metabolites is a very, very, it's a very extensive term, because secondary metabolites include lot of chemicals, which are produced by plant as secondary as a byproduct of their primary metabolism, mostly as to counter, or as a means of defense mechanism, basically. In case of angiosperms, if you see, these defense mechanisms are being met by essential oils, or by flavonoids, by terpenoids, by alkaloids, which are being produced by the plants. And man, to his own use and wisdom, uses these secondary metabolites to exploit these secondary metabolites for its own benefit. And most of these secondary metabolites have been found to be medically or therapeutically very, very important. And besides, since I was teaching gymnosperms, I'd also like to highlight that the gymnosperms also have the capability of producing these secondary metabolites in terms of you see, gums and raisins and so on. And these gums and raisins are equally therapeutically important as in case of angiosperms, where you find alkaloids, etc., which are in abundance. And when we talk about large-scale production of bioactive compounds, in plant tissue culture, there are several methods that can be employed, for essential oil-yielding plants or for even alkaloids. Since I'm talking about the secondary metabolites right now, secondary metabolites will have two groups. One, of course, is your alkaloids, which people have been attempting by and large in plant tissue culture, and they've got several very, very happy results in them. However, essential oil-yielding plants pose to be a little problematic as far as in vitro culture is there. However, the methods that are employed are similar. In both the cases, one is your micropropagation, the second is the hairy root culture, which is of course through the genetic transformation, and the third, of course, is your cell suspension culture. So micropropagation could be similar, producing plants of its own type. Sometimes you do come across, when you go through callus cultures, you do get soma clones, as we got in our case. We got several soma clones which were very, very good agronomically, and also had a share of very good oil yield, quality wise as well as quantity wise. So sometimes my simple micropropagation procedures also help to produce plants yielding good quality of essential oils / secondary metabolites. However, hairy root cultures are important in plants like where the roots are the prime source of your essential oils or your secondary metabolites. What happens is that the plants, usually these medicinal aromatic plants, have shown that the roots are very, very rich in alkaloids and essential oils. In normal cases, what people do is they harvest the entire plant, and then of course they distill the oil, or they extract the alkaloid of importance, from hairy root, from these normal roots. However, hairy root culture, gives an alternative source of production of these without, destroying the plants. And of course, you can upscale the amount of, or you can upscale the production of bioactive compounds, by using what you call as the bioreactors and so on. And last but not the least, sometimes you also have cell suspension cultures, wherein you can produce these essential oils. And there have been quite a couple of reports, where plants have been, produced. The secondary metabolites have been produced using elicitors, precursors, or giving them an abiotic stress. Elicitors like jasmonic acid, salicylic acid, they have been very, very helpful in upscaling the production of the bioactive compounds, especially in terms of essential oils. All right. So essential oils can be defined in several terms. Some define it as just a product, obtained from a natural raw material of plant origin by steam distillation. They could be obtained from any part of the plant, they could be from the leaves, they could be from the root, they could be from the flowers, they could be from the peel of the fruit, as you find in most of the citrus cases, citrus fruit cases. Or it could be done by dry distillation, okay, where these phases can be separated. They can also be defined as the volatile substances, which of course are aromatic most of the time, and they are generally extracted by hydrodistillation. They are complex mixtures of compounds because they are basically terpenoids, and terpenoids could be monoterpenes, they could be diterpenes, they could be sesquiterpenes, depending upon the type of plant. And they could be, they are usually, in case of plants, they are usually present in plants as in glandular hairs, which is most common, because they are called as trichomes, all right, or they could be modified parenchyma cells, or they could be small ducts, which are present in case of Umbelliferous rapaceae, which is like Vitae, or they could have been in passages, as in case of Pinaceae, lysogenous or schizogenous cavities, in case of Pinaceae and Rutaceae. Most of the families, in fact, couple of families, most, I shouldn't be saying most, but couple of families show the presence of essential oils, such as Pinaceae is one of the most important, which is, of course, belongs to gymnosperms. Then you have Lauraceae, you have Rutaceae, you have Apiaceae, you have Lamiaceae and Asteraceae, very, very rich in essential oils. And they have been used from times unknown, in several areas, and most recently essential oils are being used in aromatherapy, which is such an upcoming branch of every day-to-day living, because aromatherapy has taken over any other therapies so far. So essential oils have formed, and of course they're being used in medicinal purposes for making of perfumes, etc. So essential oils have been a part of us from times unknown, and of course now they are gaining a lot of importance. In fact, if you go to anybody's house nowadays, you usually find that these they have these perfumers, or they have these diffusers, which are very, very on, and you have essential oils being put, and creating those aromatic environments even in the domestic areas. So essential oils are generally formed, by three main pathways. One, of course, is your mevalonic pathway, the shikimate pathway, all right, and the other one, of course, is your pyanoid pathway, IPP. So basically these pathways lead to several stages, there are several steps to the formation of these essential oils. And these pathways, basically, if you elucidate these pathways, then it becomes easier to produce essential oils at or cut down these pathway at any point of time, so that you can have the produce of the component that you're looking for, the or for the quality of the essential oil. For example, if you talk about the pelargonium oil or the geranium oil, the linalool content, which is one of the component of the oil, is very, very important. And when one looks into the quality of the oil, we look into the geraniol and the linalool content of the oil. If it is high in geraniol and linalool content, the oil quality is considered to be very, very strong and very, very good. So therefore, these pathways are important to elucidate once we are targeting plant tissue culture for essential oil, because once we know these metabolic pathways, it is easier to manipulate these pathways for the production of the good quality oil, and maybe upscale the kind of component that we are looking for in the oil. And these pathways, as I told you, they can be manipulated by the use of enzymes, by the use of inhibitors, and so on in tissue culture. Many researchers, in fact, are being oriented towards this. And some of the common essential oil bearing plants, which are now the thirst area plants in this area, are one is, of course, pimenta. All of you must be knowing. There is APM. Umbelliferae is very, very rich in essential oils. So many members of Umbelliferae would be a part of it. And then you have Alpinia galanga, which is, of course, Zingiberaceae. And the Campharia also is a part of this family, which is your black ginger, the galanga ginger that is being talked about nowadays. And then you have citrus or the Rutaceae family. So most of these plants are being worked out, and people have worked them out in tissue culture. But while you are working plants in tissue culture, which are rich in essential oils, there are certain problems that one needs to, address as far as plant tissue culture is considered. Number one, these essential oil bearing plants generally are very, very pubescent. All right. When they are pubescent, then of course the hairy surface makes them very difficult to sterilize or get an aseptic culture in tissue culture. These trichomes, are places wherein the surface sterilization is very difficult to achieve in case of essential oil eating plants. And the second point is that the light intensity tends to play a very important part when you talk about plants of bearing essential oils in tissue culture. So two things which are difficult to achieve when you are talking about essential oil yielding plants in tissue culture is, one is your light intensity, and of course your surface sterilization. And these two things have been attempted time and again, though people have attempted a number of researchers in this area, but come to think of it, not many have come as a field trial or on a commercial basis. So these are few examples where, elicitation has happened in adventitious roots of Artemisia. Artemisia is a very important medicinal plant, and artemisinin is the alkaloid, but people have also worked on the essential oils of Artemisia, and they have seen that the elicitation works in the adventitious roots. So they have gone to produce the roots instead of focusing on the production of plants in case of Artemisia, because direct production would cut down on the labor cost and would also cut down on the time period we are looking for the production of essential oil. Similarly, production of essential oils in vitro in Caryopteris species has also been attempted. These are the few examples which we can quote, where essential oil has been attempted in case of plant tissue culture has been attempted in case of production of essential oil. So when the talk began, sir said if I could see the geranium plant. So this was one of the biggest achievement we had, where we could prove that there could be a qualitative improvement in the plants. And these were the genetic transformants using an agrobacterium rhizogene strain, an A4 strain. And Suchitra ma'am has been a part of this paper also, because she was key. She was my mentor in the department as Professor Banerjee. We were talking in the morning about it. And if you see, sir, we had tried to produce hairy roots, because generally pelargonium has essential oils in the leaves. And we went on to produce essential oils in the roots of the plant. And these roots were not normal roots, they were the genetically transformed roots. So we only used wild strains. We did not use any gene, we did not incorporate any gene, but we could get a very improved essential oil quality in case of the roots of geranium, the genetic transformants of rose-scented geraniums. And these plants showed considerable improvement over the existing normal type of plants. And one of them, of course, this was the genetic transformant plants. We had also worked on the soma clones of these rose-scented geranium, which were, of course, non-transformants. And even they had produced better quality essential oil, whether that the hormonal supplements do the job, or whether it's the controlled environmental conditions that they get in vitro, that the production of essential oil is of a better quality, and quality, of course, both the yield as well as the composition, both were better in case of the soma clones. And they were stable for years, because we cultivated them for almost 5-6 years, and they were stable for those many years. I was talking about the limitations of in vitro multiplication and regeneration. So as we were talking about the light intensity, has to be very, very well regulated when we are talking about essential oil production. And usually if you see in vivo, the essential oil plants generally are harvested in the early period of a day, because that is the time and it is one of the best quality. And of course it is, the yield is also high, because the sun is not so strong, because these oils are volatile and they don't tend to diffuse once the sun is very strong. So this is one very important point. While the growth of the plant and the light intensity has to be managed, the second, we were talking about explant sterilization, and the explant sterilization in case of essential oil yielding plant is very, very difficult, because they generally have a lot of infection on surface. Though of course the plants are antimicrobial, their products are antimicrobial in nature. However, the surface sterilization or the explant sterilization is difficult to achieve in vitro as far as essential oil plants are concerned. And then of course there are a number of hormonal imbalances that one comes across, because we generally tend to supplement them with auxins, cytokinins, and gibberellins. And these imbalances have also, we've seen in plant tissue culture, that they do happen in case of essential oil yielding plants, probably since they are very high in secondary metabolites or whatever. And the browning of explants occurs very regularly, probably because of the polyphenols and so on which are present, because they're equally rich in alkaloids as well. And of course, construction of tissue culture laboratory, which is a major task as far as essential oil yielding plants are considered. All right. Now, when we are talking about this, we have seen that in tissue culture, sometimes there is somatic hemorrhagenesis which is not achieved. There is no extensive proliferation, and of course that results in the reduced oil yield of the plants. The present, in fact, I should be saying the modernization of plant tissue culture moves towards the metabolic engineering, which is being harped upon nowadays. And the most important part is that now we are trying to do metabolomics rather than do just simple plant tissue culture, which is a very, very broad term, because metabolic engineering would then only focus on the pathways and will be very, very specific with the use of enzymes, elicitors, etc., wherein one can change the entire pathway, or obstruct the pathway to his or her own benefit. And this is exactly what has been happening in very several plants. One is your Artemisia, which has been very, very worked out. Then you have plants, horticultural plants like strawberries and so on, as it's been shown. So you have these pathways, as you can see, and you can see that these pathways can be obstructed at places to produce the alkaloid of their desire. And enzymes which can be manipulated, all right, include your fatty acid synthesis, the thioesterases, the desaturases, the sterol, these acyl transferases, the hydroxylases, and so on. And these have been reported and how they have been manipulated for the production of essential oils, and of course this thing. And peppermint is one of the very important plant, mentha, where essential oil yield and composition has been considerably achieved by metabolic engineering, where they have been able to produce quality oil in very limited time period directly through cultures and so on. The latest that has come up, in the and tissue culture area is the use of nanotechnology. And nanotechnology is one where the nanoparticles can be used in production of essential oil yielding plants, and obviously the upgrading of the quality of essential oil. The two important factors that work as far as nanotechnology is considered in case of essential oil yield plants. So one is that the surface sterilization using these nanoparticles has been very, very helpful in explant sterilization in case of essential oil yielding plants, which of course was difficult to achieve in normal plant tissue cultures, because most of the time they were herbaceous members, and if you over sterilize the plant material, generally it gives away. So nanoparticles have been able to achieve that, and somehow they have been able to generate that ROS, because of which the secondary metabolite production gets upscaled, or it increases, in case of essential oil yielding plants. So nanoparticles, and a couple of reports which have been coming in while they'll be using silver nanoparticles have done a tremendous job in case of plant tissue culture area as far as essential oil yielding plants are considered. And I believe and I hope that in coming few years, nanoparticles become the answer to it, of course, with their own disadvantages of causing toxicity. However, there have been very positive reports about nanoparticles being used in the production of essential oil yielding plants in plant tissue culture. And of course, while at the same time, they have been able to achieve a good quality and quantity of oil. And as far as the future prospects of this is considered, we can talk about it, and we can say that yes, the new age revolution is the nanomaterials which can scale new heights in plant tissue culture, of course this time for only essential oil yielding plants, and use of elicitor molecules for upscaling production of desired components and product of essential oils. They could be physical by creating radiations, by creating heat treatments, or they could be chemicals, as I talked about earlier, or which is the third line of elicitors, which are the biological elicitors. And people have seen that these fungal elicitors have been very, very candid in achieving a production of good quality essential oil yield in tissue culture. So obviously one of them is Penicillium, Glomerella, Candida, you have Basidium, you have Phylostricta, and you have Colitotrichum. So all these fungal elicitors have also been very helpful in achieving essential oil yielding plants in plant tissue culture. And with this, I would like to stop my presentation here and say a thank you to all of you. It was a very, very basic presentation because I didn't know I'll be addressing what kind of audience, but I believe I've been able to, convey some bit of part as far as production of essential oil and oil yielding plants are considered in plant tissue culture. Thank you so much, sir.
Thank you, madam. So we have listened a very informative on essential oil yielding plant tissue culture. And although it is not a very complicated research work, but what you have presented is just outcome of research work in a very lucid manner for the students, and for it is a class material type of thing. If I am a student, so I would have been really happy to listen your lecture as a student. So this is so madam, you have described the very basics of tissue culture, what is the purpose of it, and these are for students. So any student in any tissue culture class, these are very basic, so these are needed. And then what are the stages of micropropagation, organogenesis, use of hormones, and somatic embryogenesis. Then methods for large scale production of bioactive compounds. How can the different methods, what is the method for bioactive compounds that be generated, and as these are terpenoids, essential oils are different types of secondary metabolites, how they can be generated, how they can be produced. Then you have entered into your own field, and these can be generated using micropropagation, hairy root, where it is root-based essential oil, and then cell culture technique and elicitation. You have given importance of elicitation by different types of material, by abiotic stress or different types of fungal stress. At last moment, you have spoken about the importance of elicitation by different types of fungal material. So these are also very important. And then you have what could be the different types of source, what could be the occurrence in different parts of the plants which could be used. And then the biochemical pathway, which is the elucidation of biochemical pathway. You have talked about the importance of elucidation, because at different points of this biochemical pathway, I do not know, this is one of my questions. So after this session, after this part of my just appreciation and what you have told in your lecture, so it will be open for discussion. All the keynotes are normally not, questions are not accepted, but if you allow, then we'll ask questions. Anyway, so the biochemical elucidation of biochemical pathway is very important. Then manipulation to obtain specific compound by manipulating this biochemical pathway at different steps, by genes, by enzymes, by manipulating this, by generating mutants. Or I do not know whether you have generated any mutants, or there are examples of generation of mutants. And then improvement of quantity. Quantity is very important. So it is the commercially viable thing. So quantity is really very important. And then qualitative improvement, which you have done this rose-scented geranium plant. And then establishment in the field condition. When we'll cultivate, we will obtain it from plant material. So naturally, it is just from laboratory, from test tube, it is not possible. So unless we put it in the field, so it is of no use. So establishment of the field condition, its stability, these are very important. And so you have pointed out these importance also. And then what could be the different types of constraints, and then further improvement can be done by metabolic engineering. And also you have pointed the biotic elicitation and all those things in your lecture. So I think audience, I do not know how much of, including this YouTube live streaming, I hope a good number of audience is there. And definitely, I believe that the audience will be tremendously benefited from your lecture. If anyone is planning to do any tissue culture of any kind of plant, not only in essential oil plants, so they will be benefited from your lecture. So we must appreciate you a lot for giving this very important, very lucid lecture on this field of essential tissue culture of essential oil yielding plants. And here Professor Nirmal Banerjee is an expert in the tissue culture field. I hope he will also be happy in listening to this lecture. So as a general person, general botanist, general biologist, so I am very happy being the chair of this today's session. So I must appreciate from the Department of Botany. I like to extend my heartiest thank to you, madam, and I request Professor Nirmal Banerjee to comment on this topic.
First of all, thank you, Professor Naran Mondal, my ex-colleague, for asking me to interact with Professor Gauri Saxena. I think we had a small discussion in the morning, and I know she has worked for a very long time in CMAP in association with very eminent persons in this field, including one of my very good friends, Suchitra Banerjee. So that's okay. And so far I have been following her lecture, and it is, first of all, I will congratulate her for giving such a wonderful, lucid talk, which I think a small audience, there are 22 or say 25 persons actually listening to this, but they have been tremendously benefited because this is a very new field. And one of my good friends, Adin Purna Mitro in Kharagpur IIT, he is also involved in research with this kind of essential oil production in vitro system and its augmentation, etc. So I don't have to say anything, but I will just have a very curious query about this, that have you detected any new compound as essential oil during your study? Because quantitatively it can be improved, and the existing essential oils which are actually produced by a field plant can be taken up. But is there any, sometimes it happens that a new fraction or a new compound is actually generated during this culture process. So that gives very good impetus to this particular kind of research. So have you come across with such things?
Yes, sir. Yes, sir. Met plenty of times, sir. We've been able to, in fact, I've got number of publications also on that, on the new compounds that we come across in tissue culture. And I probably feel that happens because of the stress, that is given to them during the culture conditions. And gamma small was one of them, which we got in case of geranium. And there'd be another report very soon, which will be coming out. We found a couple of new compositions which have not been reported earlier. New compounds had not been reported earlier in selenum also. So it often happens that the quality of the essential oil also changes during culture conditions. So not only the ratio of the major components changes, but sometimes even new compounds are also added, sir. So geranium was one of the breakthrough that we got, and that is how it gave me a lot of strength to work on others. So we have been able to, report a couple of new compounds in many plants after that.
Oh, I was expecting that. That's very good. And one more thing, that when the plants are actually transferred to the field condition, especially in CMAP, it's a very, very big institute. Do those things stabilize? I mean, what you detected in the in vitro system, the augmentation, the improvement of secondary metabolite production, essential oil production, does it stabilize through generations in the field condition?
It does stabilize, sir. In fact, it does stabilize, sir. And CMAP has been having a very good breakthrough with couple of plants. One of them. They've been transferring this technology to farmers here in the plains, and they've been taking this plant material. They even provide the technology, they even provide the plant material, and these plant materials are then sent to the field for larger production. In fact, around Lucknow you'll find a belt which is full of mentha, and there's been like considerable production of mentha. The quality of oil has also improved, as well as the quantity of oil. So farmers have really gained out of growing mentha during the off-season crop times, when you don't have a wheat season or you don't have the rice season. Then they generally crop these intercropping plants like mentha and all, and they have been able to economics wise also they've been able to achieve that.
So, that is the best part of it, I think, because it has to be actually utilized for the benefit of the farmers and all these things. That is what actually we people are lacking in the eastern part of the country. I don't know, maybe these people can collaborate with NBRI or CMAP or some other institutes, because it should be like that. Otherwise, it is of no use doing everything.
It is of no use. Yes. Yes, sir. If it's only 2 laboratories, it is of no, it just remains as a report, sir. If it doesn't go out in the plains. So in
Yes,
the beginning of the lecture, sir, that is what I told you. Bringing pelargonium to Lucknow was an achievement in itself, and DBT was very happy. So it extended our, project also. And so Chitra ma'am was my mentor at that time. She was heading that project. So that is how I happened to be, working under her through and through.
Okay. Thank you, Professor Saxena. I think we can keep in touch. Although I am a retired person now, but we can always have contact.
It will be a pleasure, sir.
Yeah.
Professor Banerjee is always welcome in the Department of Botany.
Yes. That's okay. That's fine because
thank you, Nirmal. Thank you very much.
Thank you
very much. এবং আমি just now I am calling my other audience members, my colleagues and other audience members to ask any questions to Professor Saxena. So she is ready to answer.
Professor Mondal, myself, Professor Rahman, I am having one query from our speaker, Madam, Professor Gauri Saxena. So one thing is that have you any idea regarding the change in essential oil content, plants from the high altitude and plants from the plains or the low altitude? Is there any change in the secondary metabolite content, rather the essential oil content of the same plants, your pelargonium or rose geranium, Madam?
Right, sir. So when we got this plant, the biggest challenge was to actually grow this plant. So obviously, when there was this challenge, sir, it also affected the quantity of the oil in the plains, sir. And that is what we were trying to attempt, sir. But we were able to achieve not only in pelargonium, but a couple of plants. It takes a little bit of time, sir. It takes a little bit of time means, I'm looking at a period of 5 to 6 years, sir. If I'm talking on those lines, because there have to be continuous field trials, there have to be supplementation of the soil.
Sorry for my query is not like this. Thing is like that. Generally, what happens in so many plants, the altitudinal effect is there in production of the secondary metabolites, right?
Yeah.
So, like I'm remembering the geranium. Sorry, I'm forgetting one temperate herb. Anyway, so in this context, say for example, I'm just remembering the cinchona plant.
Okay.
Cinchona plant, it is having immense effect of the altitude on production of the quinine. In the certain altitude, the cinchona plants, they are producing quite good amount of quinine. But when the same plant is grown on the plain, then the quinine content is nearly zero, or even sometimes it is zero. Right. So in this regard, my query is that as because you work on that temperate herb, that pelargonium, so is there any change in the, have you studied or have you seen any work in this regard, the altitudinal effect on the secondary metabolite production in this temperate herb, that is the pelargonium? You have worked.
So there has to be, it's a very natural process, sir. There has to be an altitudinal, effect on the production of secondary metabolites, because as you go higher on altitude, sir, the similar thing had happened in geranium also. We did not get that kind of quantity we were looking for in plains. However, we had to work for that. So that is exactly what I'm trying to tell you, so that we had to work to produce that kind of quantity of oil, in vitro, so that we could achieve the same quantity even while we are growing plants in the plains. And that was the entire purpose, of bringing the plants to plains. If we don't achieve the kind of quantity that we are getting in high altitude, the entire purpose gets defeated.
That's the point. That's why this is a very nice area of work. So is there any altitudinal effect?
Sir,
so
there is.
One very common example is there, Darjeeling tea. We are getting the flavor in
right.
grown in Darjeeling, and if we grow it in plains, So if you grow in Santiniketan, there will be no flavor.
No, there'll be no flavor.
So things like that, but it is not happening in all the plants actually.
Yeah.
In
That's
some
why
plants,
That's why madam is getting the aroma here in plains in Lucknow.
Right.
so one thing, madam, that you told in your deliberation, that the intensity of light.
Yes, sir. Intensity of light, sir. It is directly related to the altitude too, because the intensity of light is pretty high as you go over, in the altitude. So when you come down, the light intensity, of course.
So in tissue culture, I have gone through so many literature where in tissue culture conditions, light intensity plays a very important role in production of the essential oil. So from your experience, most probably you have also experienced the same thing, the intensity of light in vitro condition having immense role on production of the essential oil here in your plants.
Absolutely, sir. Absolutely.
Right.
Absolutely.
And the last point query is that regarding the nanoparticles, right? Regarding the nanoparticles, so how nanoparticles influence the growth of the plants in vitro condition?
So there are a few reports, because obviously it's an upcoming branch. So I would not say with a lot of conviction, but there have been reports where nanoparticles are one, which I talked about, was the explant sterilization is a very, very important part, because they're very pubescent plants. Most of the essential oil eating plants are very pubescent, they're very hairy, with a lot of trichomes, sir, and that is why they hold a lot of microbes, in between, and that is why surface sterilization.
Sorry for interfering again. My point is that I am stressing upon the importance or the role of the nanoparticles, as you told.
Yes, sir.
Nanoparticles. So what probable role does play the nanoparticle?
So number one, it helps in your surface sterilization during plant tissue culture, which is difficult to achieve. Number two.
I am interrupting. I am interrupting. Actually, this thing, same thing is done by a number of workers in endophytes also, where the use of nanoparticles, once the nanoparticles are used, these nanoparticles, in case of madam, the nanoparticle, they destroy the microorganisms present on the plant surface. And their broken products act as elicitors. And these elicitations, probably, I do not know in your case, but in many cases, these broken products of microorganisms do the elicitation function. And this elicitation triggers the production in higher amount.
Number two, it also helps in production of ROS, activation of antioxidant enzymes, and even regulation of specific genes. So these also help that way, in production of the secondary metabolites.
Right. That may be, madam.
Yes,
that may be also. But what Professor Mondal told, that is also one of the
That is also one of the points.
Maybe.
That is also one of the points.
Right. So in this way, thank you very much for your nice deliberation and having good comment upon your work specific on the medicinal essential oil yielding plants and their tissue culture, secondary metabolite production. Thank you very much, ma'am.
Thank you so much.
Okay. Thank you, madam. And we have to maintain the schedule. So if there is any question, so there is a contact number of Professor Gauri Saxena is there. So anybody is willing to communicate or interact with the madam, so he or she can write to madam directly. Madam is willing to answer all the queries. So right now, I would like to thank Professor Gauri Saxena and the organizers for allowing me to chair this important session. So it's a very good academic discussion with Professor Banerjee, Professor Chaudhary Habibur Rahman, and I hope the questions are common to many of the members of the audience. Professor Gauri Saxena has interacted very well. He has presented very well her topic. So I thank again Professor Saxena and the organizers. So now it is over to organizers.
Thank you, Gauri. Thank you very much for your nice presentation. And Naranda, thank you very much for chairing the session. Thank you. So we are moving to our next session.
You can show the certificate at some point.
Oh, yes. Just a minute. Origin certificate.
If you
Wait.
so you show it later. After.
Yes, I will show it later.
So
Shekhar,
now,
start. I will.
please stay with us. We have a lot of good plenary lectures. And please say Professor Nirmal Banerjee is there, Professor Rita Kundu is there. And so in the next few lectures, there are very good lectures. So these are the very worthy to listen the lectures. So please stay with us, and you please call your friends to come who are still not ready after this winter morning. So please.
Thank you, sir. Now it's time to start the second lecture, plenary lecture. So I would like to request Dr. Hemagupta Joshi, Department of Botany, Visva-Bharati, and Dr. Orunima Bishas, Department of Microbiology, Raidighi College, to chair the session. Ma'am, please.
Thank you. Good morning. Good morning, Dr. Joshi.
Yes, good morning.
Hello, am I audible?
Yes, you are audible.
Yes.
Okay. Dr. Joshi, will you do the honor of introducing this session's speaker?
Yes.
Okay, thank you.
Am I audible?
Yes, you are.
I don't know why my video is not coming. Anyway, good morning. It is my pleasure to chair this session. And in this session, we have a plenary lecture by Professor Rita Kundu, Center of Advanced Study, Department of Botany, University of Calcutta. And she will be speaking on Phyllanthus amarus, a potential anti-cancer herb of India. Let me give her a brief introduction. So Professor Kundu, she has graduated from Bethune College with honors in botany in 1985 and then masters in botany with a specialization in cell biology and genetics from University of Calcutta in 1987. Then she received her PhD degree in botany from University of Calcutta in 1999. She worked as a visiting scholar in Heidelberg, Germany for a brief period. She had teaching experience, undergraduate teaching experience from 1997 to 2004 in Lady Brabourne College and postgraduate teaching experience in the Department of Botany, University of Calcutta since 2004 till date. There are two areas of research interests of Professor Kundu. First is the cell biology of cancer cells with target to induce cell death in the cervical cancer cell lines using traditional medicinal plants and other organic sources, and to identify the bioactive compounds and their mode of action in regulating cell proliferation. Second area of interest, of research interest, is the phytoremediation and abiotic stress biology, with interest in identifying the native plants having potential capacity to remediate the contaminated soil and to study their stress responses and detoxification mechanism. Actually, her prime interest is to identify the metal accumulator crop varieties of rice from West Bengal. She has done immense publications in many national and international journals like Journal of Biomolecular Structure and Dynamics, Plant Physiology and Biochemistry, Scientific Reports, National Academy Science Letters, Ecotoxicology and Environmental Safety, Chemosphere, Journal of Plant Growth Regulation, and Free Radical Biology and Medicine, and so on. And she is life member of many scientific bodies like Society of Plant Biology, then Botanical Society of Bengal, Probir Chatterjee Research Foundation, and Archana Sharma Foundation of Calcutta, and Indian Science Congress Association. She has been a review editor in Frontiers in Nanotechnology. She is editorial board member of Journal of Botanical Society and has reviewed in many important high-impact journals like PLOS One, Helion, Journal of Plant Growth and Regulation, Cell Death and Disease, Ecotoxicology and Environmental Safety, Natural Product Research, International Journal of Phytoremediation,
Plant Gene, Frontiers in Nanotechnology, Proceedings of the National Academy of Sciences, India. Okay, with this brief introduction, I welcome Professor Kundu to present her plenary lecture. Now over to Madam Professor Rita Kundu. Ma'am, you have 30 minutes, please.
Thank you. For your kind introduction. Is it visible, the slide?
Yes, ma'am.
Yes, the slides are visible.
So the full screen is visible. Okay. The topic of today's lecture is Phyllanthus amarus, a potential anti-cancer hub of India. From the very first day of the civilization, we have seen that plants are providing us several things for our shelter, for our clothes, for our food, fodder, perfumes, for everything. We are depending on oxygen of the air, which also produced by the plants. And nowadays, we are also depending on plants for fuels. So we can see that plants are associated with us tremendously. But my interest is plants as the therapeutic agents. In Indian subcontinent, we have seen that the study of Ayurveda, that means the science of longevity, preached and practiced by Charuk, Shushruta and others around 600 BC, and they have described the curative properties of plants. And according to who, we have seen that about 80% of world population from developing countries that depends on plant-based medicine. About 25% of the drugs prescribed worldwide come from the plants. Of these 52 drugs considered as basic and essential by WHO, 11% are exclusively of plant origin, and a significant number are synthetic drugs which are obtained from natural precursors. And this is the data which shows that the number of plants found worldwide and the medicinal use. We can see that there are around 17,500 plants among these 250,000 plants which are found worldwide, and which is a greater share of 7%. So India, we can say that it's a rich repository of plants, and this is only about the angiosperms. And other than angiosperms, there are gymnosperms, pseudophytes, bryophytes, lichens, algae, fungi, and other plant members. And in the Indian biodiversity, we can see there are 17,000 to 1,800 species of flowering plants, and about them, the 7,000 to 7,500 species are of medicinal uses, and 242 species are consumed over 100 metric tons, and 1,178 species are in trade, and the exporting value is according to 1,34,500 metric tons, and the domestic consumption is 1,95,000 metric tons. So this is a huge amount of plants which are consumed and exported by us. Now, our main concern is to revisit Phyllanthus amarus. And this Phyllanthus amarus plant is a small herb of Euphorbiaceae family, and this is commonly known as stonebreaker, or in Bengali it is known as bhui amla. Traditionally, the Phyllanthus species is Phyllanthus amarus species is used for several purposes: nephroprotective, hypoglycemic, antimicrobial, spasmolytic, diuretic, hepatoprotective, antiviral, antinociceptive, antioxidant, and immunomodulative. Traditionally, for all these, mostly to cure the intestinal and liver problems, they were used. But later it was found that they can be used for antimicrobial, antiviral, and antioxidative roles. But we just want to revisit this plant for its anti-cancer activities. We just wanted to see how much this plant can be effective in treating cancer, and if it is able to show any anti-cancer role in vitro, then we just wanted to see what is the mechanism or what is the pathway by which it can induce the cell death. Now you can ask me, why cancer? Why cancer? Because in this year 2020, not in this year 2020, according to Globocan, it was found that there are 19.3 million new cancer cases and 10 million cancer deaths, and this is irrespective of sex and age. So you can easily see that there is a lot of cancer for breast cancer, lung, colorectal, and prostate and stomach cancer. They are occupying the main share, and esophagus, cervix, uterine, and some small cancers, they are occupying the 42.9% cancer cases. And the mortality is the maximum mortality, 18% mortality is caused due to lung cancer. And then there is colorectal cancer, liver cancer, and stomach cancer, and breast cancer. So we can see the appearance of new cases and the mortality incidences are very high even in 2020. And by 2040, it was predicted that 28.4 million new cases and 13 million cancer deaths are predicted. So this is a huge number. And cancer deaths are second to the death next to cardiac arrest. And this is the estimated 8 standardized cancer incidences in case of male, where we can see that the lung cancer incidences are highest, and then prostate and colorectal, which is followed by stomach, liver, bladder, and other cancers. And in case of female, we can see that breast cancer is occupying the maximum incidence rate, and followed by colorectal, lung, and cervix, uterine, occupying the 4th position. But this is an overall picture of these cancer cases. We are more interested about the female cancers, and where we can see that the incidence rate and mortality is too high, and in case of incidence and mortality, cervix, uterine occupying the 2nd position. Here you can see that the new cases in 2020 is 614,000, and death is 341,000. So it is too high. And not only that, we can see that the incidence rate is high in the low human index countries. That means the countries where the economic growth is much lower, and the countries with the high development index, that means countries with HDI, the breast cancer is the major cancer. So this is the blue region depicts the incidence, and the red one is the mortality. So we are more interested about the cervical cancers, and we can see that the Eastern African and South Asian countries are showing greater incidence rate and greater mortality due to cervical cancers. So the causes of cervical cancer, we have found that in 70% of cases, the HPV or the human papilloma virus infections are associated. These are the risk factor for HPV infection because they are rarely or never screened in the low HDI countries. HPV persistence, immunosuppression due to HIV or high-dose steroid use, history of lower genital tract neoplasia, increasing number of sexual partners, early age of sexual activity, infection with chlamydia and possibly herpes simplex virus, tobacco smoking, use of oral contraceptive for more than 10 years, and other things are full-term pregnancies and malnutrition. So these all added the risk factor for HPV infections. Now, this is the structure of the HPV, the human papilloma virus, and this human papilloma virus, it shows the surface pentamers, which are drawn here by blue. There are lots of human papilloma viruses, more than 100 strains, and some of the strains are harmless. They are just causing warts, skin warts, but some of them, they can cause cancers to humans, and these are known as HR type or high risk type, and they not only able to cause cancers in female body, but at the same time they are responsible for causing oral cancers, penile cancers, head and neck cancers in male members also. So, if I want to understand the mechanism of cell death, then I have to know about their integration and mechanism of cell proliferation. So when the HPV genome is integrated into the host cell, we have seen that there are several early genes which are depicted by E1 to E7, and there are two late genes which are known as L1 and L2. So by this way, the E1 and E2 here, the genome is open and integrated, and once they are integrated, they just hijack the host genome genes PRB and P53 by the two oncogenes, which are E6 and E7, and then they induce cell proliferation. So when the cell is under the control of HPV, what may happen? We generally know that the cell is regulating its cell cycle by the PRB genes. So when the cell is not ready, PRB is blocked and blocking the E2A, and the cell cannot enter into the cell cycle. And at the same time, if there is any DNA damage, then the P53 produces P21, and that arrests the cells at G1 failure. So when the HPV enter within the body, what happens? This HPV E6 binds the P53 and blocks its activity. So it cannot produce P21. So irrespective of the DNA damage, the cell cannot produce P21 and cannot arrest the cell into the cycle phases. So with the damage, the cell can proceed through the division and ultimately causes oncogenesis. And at the same time, the HPV E7, that also binds the PRB, making it non-functional. As a result, the E2F can trigger the cell to enter age phase prematurely. So we can see that the P53 and P21 as well as PRB can be hijacked and blocked by two oncogenes of the HPV E6 and E7. So if we want to induce cell death, then what we have to do? So we can see if there is irreparable damage within the cell, then not only the p53 arrests the cell into cell division phases, but at the same time produces some apoptotic genes which are BAX and NOXA, and it can block the BCL2, which is responsible for cell survival. And this BAXA and NOXA, they binds on the mitochondria and make some force on it, and as a result, some apoptotic substances like cytochrome C and other proteins come out of the cell and can causes cell death. So this is the mechanism of cell death. So we just wanted to see how our plant extracts fit into this model. And at the same time, we also see that when the HPV infects the keratinocyte, then what happens? The cells secreted some substances which are known as ET1, and the cell itself also have their receptors which are known as ETBR. So the ET1 binds with this ETBR, and these ETBR, as they accept this ET1, which is a paracrine signaling molecule, or you can say an autocrine signaling molecule. So this signaling molecule induces the cell to synthesize some vasoactive substances, VEGF. So the new angiogenesis occur, and at the same time, they secrete some substances which help the cells to transform them from the epithelial cell to mesenchymal cell. So there is an ENT pathway, and due to this ENT pathway, we can see that this cell entered into the blood vessels and then moved to other places, and this is known as invasion and migration, and they can go to other places and then they can cause proliferation there, and in this way malignancy occurs. So we can see that how these human papilloma virus can induce malignancy within the human cell by making the p53 ERB non-functional and making the vasoactive substances by the keratinocytes. Now, the standard treatment procedure involves surgery, radiotherapy, hormone therapy, and now the treatments of the 20th century includes chemotherapy, immunotherapy, and targeted therapy. But all these therapies have severe side effects like immunosuppression, loss of appetite, loss of hair, loss of fertility, delirium, peripheral neuropathy, bleeding, and anemia. So our target is to find some active biomolecule with selective cytotoxicity with either no or lesser side effects. And for these, we are trying to revisit Phyllanthus amarus. We have already seen that there are certain preliminary work by other researchers on K562 cells, ascites carcinoma in Swiss albino mice, on GLA cells and Neo cells and PC3 cells and NCF cells. Some of the workers, they have tried the extract, or some of them have tried to isolate the pure substances, bioactive compounds from these phyllanthine, hypophyllanthine or neranthine, and they tried to treat it. And in all cases, they have seen that the cells were induced to death. So we can see that these substances are a degree of cytotoxicity. So to evaluate the anti-proliferative properties of P. amarus, organic extracts were studied on a panel of cervical cancer cell line. For that, we have used HeLa, which is HPV 18 positive cell line. We have taken SHIHA, HPV 16 positive cell line, and C33A, HPV negative cell line. Among these, we have seen that the prevalence of HPV 16 positive cervical cancer are more in Indian subcontinent. So we have extracted our plant and taken 3 fractions. One is hexane, one is chloroform, and the other one is aqueous. And we have done the entity assay to see the cytotoxicity. And you can easily see from this graph that the chloroform extract shown better results on HeLa, Shiha, and C33. And this is the normal healthy cell line, the NIH 3T3, which we have taken to see what is their effect on the non-malignant cells. And this clearly shows that the extract is selectively cytotoxic. It has effect only on the cancer cells, but it has no effect on the NIH 3T3 cells. So this is the IC50 dose of Shiha cells. So we can see that the Phyllanthus amarus chloroform extract at a dose of 50 mg per ml can inhibit the concentration of Shiha cells at 50%. So this is the best concentration of this extract. So this is the morphology of the cells. So this is the phase diagrams, and this is the nucleus stained by DAPI. And you can see that as the cells were treated, they show the apoptotic features. The nucleus is condensed, and the cells show some kind of bleeding, cytoplasmic bleeding, which is clear from this morphology. If we have any effect on DNA, so we have isolated the DNA, and the DNA show clear fragmentation, and this is a hallmark of apoptotic acid. So you can see that there is a ladder-like structures. So the 3 is the chloroform fraction, and the 4th one is the hexane fraction. So in all the cells, we can see that there is a DNA fragmentation, but the aqueous extract has no such effect. So we can see that in HeLa, Shiha, and C33, the Phyllanthus amarus chloroform extract can induce DNA breakage. We have studied the DNA breakage by the COMET assay, and you can see that this is the control cell, and this is the COMET, which clearly shows that only the undamaged DNA is there, and most of the damaged DNA are traveled and form a comet tail. During apoptosis, what happens? The inner membrane flipped outside, and as a result, the phosphatidylserine comes outside, which is generally present in the inner leaflet. So as the membrane flips, the phosphatidylserine comes outside, and this phosphatidylserine has affinity for the annexin, which is a calcium binding protein. So an assay was designed where the annexin is associated with the fluorophore, which is FITC. So when we adhere this annexin with the fluorophore, it is found that the cells which are annexin positive can be detected through the facts of flow cytometer. So in all these cases, we can see that this is the control cell, and this is the treated cell, and this is the live cells, and this is the early apoptotic, late apoptotic, and the necrotic. And in all cases, you can see as the cells were treated with the PAC, that means the chloroform extract of the Phyllanthus amarus, the apoptotic percentage of cells increases and the live cells decreases. And at the same time, we also studied the cell cycle by flow cytometer. In all cases, we have seen that the sub G0 phases increases, and not only that, in Shiha, we can see that there is a greater increase in G1S phase. That means that indicated again that there is a DNA damage. So we wanted to see the expression of certain genes by RT-PCR, and we can see that the expression of BCL2 BAC, P53, P21, and ET1 gene increases. Sorry, not ET1. P21 gene increases in the treated one, whereas the expression of ET1 and HPV E6 gene decreases. This clearly shows that the extract is able to induce the apoptotic genes, and at the same time it can decrease the expression of the oncogenes. And then we have tried to see the expression of these proteins through Western blot. And in this Western blot, you can see that the caspase is playing a very important role in cleaving other proteins. So cleaved caspase is found in all the cell lines. The expression of BCL2 decreases, expression of BAX, P53 and P21 increases, and the expression of E6 is decreases in all the cell lines. This is the histogram depicting the fold increase of these proteins. So what is the ROS within the cell? So we estimated the ROS generation, and as a result of ROS, we also wanted to study the loss of mitochondrial membrane potential within the cell. And it can be clearly seen that the ROS generation is increased more in all the cell lines than the control cells. So the phylum plus amorous chloroform extract were able to induce ROS, and as a result of that, the ROS which generated free radicals, and that free radicals induces certain changes, and that changes damages the DNA, and as the cell senses the damaged DNA, so it increases the certain substances, and which causes the loss of mitochondrial membrane potential. As the membrane potential is lost, so the apoptotic substances, that means the diaglo and cytochrome C, comes out from the mitochondria and causes cell death. Then we started the intracellular flow cytometry, where the cell is treated with the cleaved caspase, which are associated with the fluorophore, cleaved PARP, which is responsible for DNA damage, and the BAX, which is responsible for inducing cell death, which binds on the mitochondria. And then we also studied the BCL2 and P53. In all the cases, we have seen that the caspase and the PARP concentration, as well as the BAX, increases as we have expected, but at the same time, the expression of BCL2 decreases. Now, as the DNA is damaged, so how this DNA damage can be studied in situ? So we already studied the DNA fragmentation by DNA gel as well as by the chromatography, but we wanted to study the DNA breakage in situ. So we used gamma H2AX, which is associated with FITC fluorophore. So what happens when there is a DNA damage? The H2A is replaced by the histone. H2A is replaced by a variant, and that variant becomes phosphorylated on its gamma position, and so it is known as gamma H2AX. So we have used a specific antibody to target this gamma H2AX, and as it is associated with the fluorophore, so we have been able to pinpoint the position of this gamma H2AX loci. And as along with that, we have studied certain proteins which are associated with the DNA damage. That is the phospho-ATR, which become active during the DNA damage, as well as the checkpoint kinase 1. So in both the cases, we can see that the expression of phospho-ATR and phospho-checkpoint kinase 1 gene increases. And this happens in HeLa, this happens in SHIHA, as well as in C33A. That means irrespective of the HPV presence or absence, our extract is able to do its activity. Then we wanted to study what is there in the PAC, and for that we have analyzed through GCMS, and through GCMS analysis we have found that these are the probable compounds. There are some squalene which are actually the precursor of several fatty acids. There are dibutyl phthalate and icosane. So several bioactive molecules are found there. Then we studied for some high molecular weight compounds present in this, and for that we go for LCMS. And under LCMS we have isolated quercetin, neranthine, rutin, flavonone, and correlatin, so many bioactive compounds. Then we fractionated these substances under column chromatography and isolated these fractions, and these fractions were found to be having cytotoxicity. And pyrazanol, benzoic acid, chrysin, and cinnamic acid were found to be the major compound isolated by this NIST library. Then from this column, we have isolated one crystal, and through the NMR study and proton NMR, we have seen that this compound is phylanthine. So this is the ESI mass. And with this phylanthine rich, which is nothing but a lignan, so with these lignan rich substances, we again treated our cells and seen their morphology under the fluorescence microscope and studied their mitochondrial potential as well as the cell cycle. And in all the cases, we have seen that the mitochondrial potential decreases and the cell cycle shows G1S phase arrest. So we found, we are now absolutely sure that this phylanthine is the major bioactive compound present in our extract. And this is the FITC assay, which again shows that during the treatment, the apoptotic, early apoptotic and late apoptotic percentage increases. This is the control and this is the treated one. And again, we have studied the genes and protein expression under the microscope by immunoblotting and real-time PCR. Then we have docked our phylanthine with 3 proteins. First of all, we studied the phylanthine with E6 protein, and we have found that phylanthine can bind the E6 protein with a free energy of minus 4.89 kilocalorie per mole, which is very high. And then we have studied the phylanthine with MDN protein, and we also find that it can also bind the minus 3.47. And when we studied the binding of P53 protein, we can see that there is a binding energy of plus 124.37, which clearly indicates that phylanthine cannot bind with P53. So, in a normal healthy cell, what happens when P53 is there? MDN binds P53 and transports it to the cytosol and cleave it. So P53 is not available, and as P53 is not, and the cell cannot arrest the damaged DNA or damaged cell. And in presence of HPV, that happens by the E6. So when phyllanthrin is present there, as phyllanthrin block the E6, so E6 cannot block P53, and at the same time, as phyllanthrin is also able to block MDM2, so MDM is not also able to bind the P53. So we can see that the P53 remains P, and as the P53 concentration increases within the cell, that we have already shown in our other experiments through real-time PCR and Western blotting. So we are proposing the model where the phyllanthrin can block the E6, and as E6 is blocked, it cannot bind the P53. As a result, the P53 concentration increases, and as P53 concentration increases, it decreases the BCL2 and it increases the BAX concentration, and at the same time P21 is also increases, and as P21 increases, it blocks the CDK. So the cells show G1-S arrays. And as BAX increases, we can see the caspase 3 is also increases, and there is loss of MMP. And as there is loss of MMP, cytochrome C is released and causing apoptosis. And at the same time, we have also seen that the ET1 is decreased, and as ET1 is decreased, so the cell proliferation is also decreased. And due to Ross generation, there is damage, and this damaged DNA again increases the P53. So this is the proposed model of our Phyllanthris amorous chloroform extract, and we have published this in scientific reports. Last of all, I would like to share a quote from Hippocrates: "Let food be thy medicine, and medicine be thy food." Thank you.
Thank you, Professor Kundu. It is a real privilege to co-chair this session along with Dr. Joshi. So thank you, organizers. And Professor Kundu, what can I say? That was such a outstanding presentation, and informative, lucid, and it's really incredible work, extensively done, mechanisms and all, everything wonderfully explained, you know. So thank you. I mean, we hear the term cancer and we are really filled with dread. We think we are done for, and the treatments are so expensive and painful with severe effects. So if a natural product like this can give a better therapeutic alternative, a targeted therapeutic effect with minimum or no side effects, and that will be such a blessing. So really wonderful work. Thank you so much for sharing this with us. And I'll request the participants, if they have any questions, kindly unmute and you can ask your question. I don't see any question in the chat box right now. So if anyone want to ask, please unmute and you can ask questions.
Thank you, Professor Gupta. Thank you for your
okay,
and
Rita
I will be very happy to answer any questions. Volume.
Rita Di, you have presented a very nice work of a plant-derived plant metabolite to combat the cancer. And just I have three questions. One is how it differentiates the cancer cells and normal cells. As you are getting your MTT result is okay, I am accepting that. But when you have shown that ETAR and ETBR, probably some these membranes which are present in this normal cell, is there any differentiation of this membrane receiving the molecule or ETBR is playing any role in this differentiation process?
The keratinocytes which are infected by the HPV secretes the ET1 and endothelin 1, and these substances are secreted only by the HPV infected keratinocytes. These are not present on other cells, and these ET1 receptors are also present on the keratinocytes. And as these keratinocytes are secreting these substances, these have some properties of angiogenesis and vasoactive properties. And at the same time, they also induce the cells from the epithelial to mesenchymal transition. And as ENT is one of the property of the malignant cells, so these ET1 induces the cells to go for the ENT transition.
Okay, ma'am.
And as these cells, other cells are not having these properties, so they are not showing this activity. So we have seen that these Phyllanthus chloroform extract can inhibit the ET1 production.
Okay, ma'am. Thank you. And one more thing.
Yes.
You mentioned ascites carcinoma, and I missed it. Whether it is your work or you have cited.
No. These are not my work. These are previous works.
Okay.
We have done only the in vitro culture.
And just one point. And
we have not
metabolite, if we apply this metabolite in cell line, or you have worked it on cell line, not on
cell line.
In vivo system, you have not worked it out.
No.
Okay.
Thank you. Is there any further question from anybody? Okay, then, ma'am, thank you once again. And let me present a small token of appreciation on behalf of the organizing committee for you.
I hope I have within the time limit. So
No, absolutely. You're absolutely within the time limit. And that was very lucidly done too within the time limit. Thank you.
I request Oroji, you please share the certificate of appreciation of the Professor Rita Kundu. Oroji,
yes, ma'am. Yes, ma'am. Just a minute.
Okay.
Share করাই যাচ্ছে।
এখানে শেয়ার করতে পারলে ভালো, তুমি ম্যামকে মেল করে দাও।
হ্যাঁ ম্যাম,
ম্যামকে মেল করে দাও।
যাচ্ছে না
মেবি নেটওয়ার্ক প্রবলেমের জন্য এখানে করতে পারছে না। হ্যাঁ, সেশন আমরা কন্টিনিউ করছি, ঠিক আছে? সো কি নেক্সট হতে আমরা যেমনি চেষ্টা করছি, ঠিক আছে ম্যাম? ম্যাম সরি, একটুখানি
no problem.
Ma'am has presented an excellent work, so we
yes. Gauridi ma'am always done a very good. I'm very thankful to Gauridi. I'm very thankful to ma'am. As a we have choose Gauridi and ma'am, and whatever the speakers, they are very share the informative lecture, and I'm from the core of the heart for their cooperation, and each they shared me each and everything in the time. I'm very thankful. I'm very thankful to all. So because without their cooperation, I think our seminar could not be success at this rate. Thank you very much. Okay.
Thank you all.
Thank you very much, ma'am. Now it's time to start the second and finally plenary lecture. For this, I would like to request Dr. Adani Lokho, Department of Botany, Visva-Bharati, and Dr. Shoumi Naha, Department of Botany, Ananda Mohan College, to chair this session. Sir, please. Ma'am, please.
Am I audible?
Yes, you are audible.
Yes, ma'am.
Good morning, all of you. Warm regards to all dignitaries, my respected teachers, colleagues, dear students and all. It is my extreme privilege to chair this session along with Dr. Adani Lokho and to welcome you all to the plenary lecture 2 on the second day of this online national seminar on cytogenetics, in vitro culture and phytochemistry of plants and microbes for sustainable use. In this connection, I would like to thank the joint organizers of this seminar, Dr. Anjalika Rai, Department of Botany, Visva-Bharati, and Dr. Madhumita Majumdar, Head, Department of Botany, Raidighi College, and all other associated persons for wonderful organization of this two-day national seminar, and also to give me this opportunity to welcome Professor Nirmal Banerjee, who has guided, supervised, and mentored many of us, and we are extremely privileged to get his guidance in all our academic endeavor till date. Now, regarding Professor Nirmal Banerjee, he graduated with Botany Honours from Presidency College, Kolkata in 1975-76. He did his MSc in Botany from University of Calcutta with specialization in Cytogenetics and Plant Biotechnology in 1978-79. He did his PhD from Department of Botany under the supervision of Professor Arun Kumar Sharma in the field of Cytogenetics and Plant Tissue Culture in 1983. He did his postdoctoral research as fellow of the International Board of Plant Genetic Resources under the aegis of Food and Agriculture Organization of United Nations in Laboratory of Tropical Agriculture, Catholic University Leuven, Belgium. And he did his another postdoctoral research training in Plant Biotechnology Laboratory, Nottingham University in 1985. After that, he served in Darjeeling Government College in 1990 and then in Department of Botany, Visva-Bharati from 1990 till he retired in 2019. He supervised 22 PhD students in Visva-Bharati and published more than 80 research papers in national and international research journals during this tenure. He acted as expert member, chancellor nominee for the selection of various academic positions in different universities within and outside the state of West Bengal. He shouldered various academic and administrative responsibilities while serving in Visva-Bharati like librarian, finance officer, director or coordinator of IQAC, chairman of admission committee, etc. So I would like to welcome Professor Banerjee for his plenary lecture on propagation and increased secondary metabolite production of Solanum erianthum D-DON through in vitro approach. Now over to Professor Nirmal Banerjee. Sir, please.
Is it okay? Am I audible?
Yes, sir.
Okay. So thank you, Shomi and Dr. Loko for your kind introduction. I'm a local person from Shantiniketan. And first of all, at the outset, I would like to thank the joint organizers of this particular national seminar, Dr. Anjalika Roy and Dr. Madhumita Majumdar. They are originally from my department, that is Department of Botany, Visva-Bharati, but Anjalika is now serving in Visva-Bharati Department of Botany as an assistant professor. And Madhumita is also a product of this university, but she is presently engaged in as an assistant professor in Raidighi College under Calcutta University. So I should pay my thankfulness to these people as well as the entire faculty of Department of Botany. All of them are my colleagues, although I am slightly out of touch now because I am a retired person, but I keep in contact because I live in Shantiniketan. So I have been listening to the lectures by Professor Gauri Saxena and my beloved sister, Rita Kundu of Calcutta University. And both those lectures are actually very, very impressive. And I was really very happy to listen to their lectures. In future, we can interact with them. Actually, today I served this department for more than 30 years. in Visva-Bharati, and I was actually heading the laboratory of plant tissue culture and cytogenetics there. So most of the time, actually, I engaged myself with the work on medicinal plants as well as some floricultural, horticultural plants, especially the orchids. So this time I have not selected orchid because I worked for more than 20 years on orchids, but I have selected a single plant, that is Solanum erianthum, which is actually a plant which is threatened and endangered in this particular part of the country. And I have chosen, this is actually the work of one of my PhD students. So I will just try to highlight some of the important aspects of her work within this short period of time. Okay, so the topic is propagation and increased secondary metabolite production of Solanum erianthum, this is a relatively less known plant through in vitro approach. So this is just to introduce you with this particular family. All of you know that Solanaceae is one of the very important families of angiosperm, consisting of large number of medicinal and phytochemically important plants. More than 2,300 species are actually available in our country. And it has got a lot of economic importance. This particular family as a whole, economic importance for its food value and pharmaceutical demands. Many plants are sources of chemical compounds of relevance in modern medicinal and pharmacological industry. A number of species of Solanum has been reported to contain a wide range of secondary metabolites. I will be mainly highlighting on this particular aspect along with the in vitro propagation part. And alkaloids, phenolics, flavonoids, glycosides, etc., they are all produced by the members of this plant. And among the plant species of Solanaceae, actually, this Solanum erianthum, which I have discovered accidentally during my visit to Hazaribagh University. I have not seen them. I have a report that in Kalyani region, this particular plant is actually growing in nature, but in Hazaribagh, I found this plant in abundance. So I collected the fruits of those plants from that part and I took it to Shantiniketan. Like another Solanum member that is called a tree Solanum, which is actually Solanum erianthum, Solanum acranthum, that I have collected from Guwahati University campus. So I established those plants also in the Department of Botany. So this is my hobby. So the Solanum erianthum was actually, I collected, I got information from the taxonomist of Hazaribagh University that this is a plant growing in that particular region, but I have not seen them in Shantiniketan area. Anyway, this is later on we have found that this is a very important medicinal plant, particularly for the production of a lot of secondary metabolites, including the steroidal alkaloid that is solasodine, which is also found in some other plant species like Solanum khasianum and some other plants. But this particular plant is rich in solasodine content. Later on, I have actually got it from the literature. So solasodine is a steroidal alkaloid obtained from this particular plant in high amount, quite some high amount, and is used as a precursor for synthesis of complex steroidal compounds used for the production of oral contraceptives. This is a very important aspect. Now, why this plant was specifically selected. Actually, this is a work I have given to one of my PhD students. So she worked on this particular, her name is Jita Sarkar. She worked on this plant extensively. And I will be trying to highlight some of the important aspects or works of that particular PhD student. Now, it is rich source of pharmaceutical constituents like flavonoids, steroidal alkaloids, phenolics, fatty acids, so on. That is actually, this is the importance of this plant, and it has got a lot of economic importance, like it is used for treatment of inflammatory diseases, burns, wounds, etc. The leaves have been reported as anti-malarial agent. Decoction of the root is used for body pain, vertigo, urinary troubles, etc. This plant contains steroidal alkaloid psilocybin, as I have already mentioned. Now, actually, what we tried to achieve in this particular.
Sir.
Yes.
Sir, can I interfere? Are you changing the slides, sir?
It's not changing?
No, we are still, at least I am still seeing the front page.
But I can see it. Is it? Okay, let me check it. Is it changing now?
No.
No, sir.
No or yes? দেখা যাচ্ছে চেঞ্জ
দেখা যাচ্ছে না
টাইটেল স্লাইডটাই দেখা যাচ্ছে
তাহলে কি করব
Please click on the second slide. Just put the cursor on the second slide.
Okay, should I
Nirmalada, enter screen
এখন আসছে
এবার দেখা যাচ্ছে
কিন্তু এটা কিন্তু ফুল স্ক্রিন হচ্ছে না তাই তো
No, sir, it's not full screen, but that's not a problem. I think it's properly visible.
It is visible. You can
Yes,
read
sir,
it.
it's
Okay.
clearly visible.
So I will stick to that.
Yes, sir.
No problem. Okay, let us go back to that one. Yeah, this is just to tell you the major objectives, actually, what we tried to achieve in this particular study. I will mainly focus on two major aspects of this particular plant, what she did, that girl. The first aspect is in vitro aspect, that includes a lot of tissue cultural areas. And the second one is phytochemical aspect. So regarding the in vitro aspect, it is development of an optimized protocol for in vitro propagation of Solanum arianthum through in vitro approach, and assessment of genetic fidelity of the in vitro regenerated plants through molecular technique. Now, in this particular point, actually, I would like to highlight that micropropagation can be achieved in various ways. In the morning, we have heard it from Professor Saxena that different routes of micropropagation are there, either direct or indirect routes. It may be through callus-mediated organogenesis or somatic embryogenesis, or it may be direct regeneration of plants, so on and so forth. There are advantages and disadvantages of each of these actually pathways or routes. So we'll come to that. And then finally, the product which you get, that is the regenerated plants, should be assessed genetically, whether those plants are genetically stable or there is some variation occurring in this plant, or they are genetically variant. So for that, actually, the present method is to assess them through molecular markers like RAPD, ISSR, etc. And the second part that will actually include the phytochemical aspects of this particular plant. Actually, we will highlight in this particular plant, a particular part, that what are the major plant parts of the plant which gives rise to, or which produces higher amount of secondary metabolite, different organs that has been studied in detail, like the leaf, like the root, like the shoot, like the flower, like the fruit, etc. And then to select out which particular part gives the highest amount of, accumulates highest amount of, or higher amount of secondary metabolite. Once you get that, you can use that particular plant for initiation of culture and then proceed. And along with that, actually, once you establish the successful culture, then that particular type of culture, which will give you the maximum amount of secondary metabolite, can be subsequently utilized for further improvement or further augmentation of secondary metabolites by application of elicitors or any other kind of media manipulation, etc. So this part actually we will highlight in the second part. And finally, we will include the hairy root culture of this particular plant for this particular part. Actually, I'm thankful to Suchitra Banerjee, Professor Suchitra Banerjee of CMAP, because she provided me the strain of that Agrobacterium rhizogenes from Lucknow, all the way from Lucknow, and we successfully utilized that, and we succeeded in getting the good result. So we'll come to that. Now the first part, the Solanum erianthum. This is just an overview, not the detailed results. I'm going to show you. So why in vitro culture is so important in this particular time? There are several advantages. The first one is economically important pharmaceutical compounds can be produced under controlled environmental conditions as per market demand. This is a very well-known fact nowadays, and Japan was a pioneering country who first started producing secondary metabolites through in vitro tissue, through in vitro culture, particularly the suspension culture and cell suspension through your fermenter system, all these things in 1970-75, like that. And you know that each and every country of the world right now, they have imposed stringent laws that the natural plant populations cannot be exploited for production of secondary metabolites or medicines. They cannot be utilized in the pharmacy, by the pharmaceutical companies, like that. So for that, actually, you have to generate your biomass somehow. So that somehow is actually, this is the only solution, is the in vitro system, where you can generate your cells and tissues and plants, which can be subsequently utilized for production of secondary metabolites. So this is the reason, actually, this particular area of biotechnology, plant biotechnology, is getting so much of impetus. So the other factors or other advantages are independent of environmental factor. This is very important. So in CMAP, you know, I've visited once or twice there. They have very big field, like your NBRI. And normally the different germplasm and all those particular important plants are actually maintained in the field condition also. But there are problems of maintaining the plants in the field condition, you know, because of these abiotic and abiotic stress factors, like whatever, the climatic factors, the insects, the pathogen, the pests, etc. So you can all avoid all these things when we are transferring the things to the in vitro system, the laboratory system, where the tissues and cultures will grow in completely defined and controlled condition. So there is no chance of any damage of the thing except some contamination. So that can be managed. Now, the other advantages are quality of plants will be consistent throughout as the products are formed by specifically grown cell lines under complete defined condition. So that is a known fact, that once you are having a defined condition where each and every component of your culture medium and the physical environment is known to you, then you can maintain that. As long as that particular condition is maintained, you get the product. Except if there is any genetic variation occurring in the form of somatoform variation, you may get some change. Otherwise, they will remain unchanged. So that can also be managed. Genetically stable clones as well as genetically variable plants can be generated. This is a very important area of this plant biotechnology, that you can, if you want genetically stable clones, clonal propagation, you can utilize the tissue culture system, in vitro system. If you want genetically variable plants, that is also possible, because if you are actually working for production of some pathogen, some disease-resistant plant or insect-resistant plant or some salinity-resistant plants, etc., you can utilize this tissue culture system without involving the genetic engineering part. So simply by growing the plants or growing the tissues in suspension condition, you can always exploit those cell lines for production of some genetically variable plants or genetically variable cell lines, which can be subsequently regenerated into plants. So this is another very big area of plant biotechnology where people are working for last several decades for production of genetically variable plants according to your desire. And finally, the biotransformation. This is a small thing related to your secondary metabolite production. So you can always convert a particular bioactive compound into another more active bioactive compound utilizing this particular cell line system or cell culture system. So that is very important thing. That is biotransformation. Like there are several examples on that. You know, digitalis purpurea, that digitoxin has been converted to digoxin, which is more active compound through this kind of biotransformation system. And for that, actually, you have to use the suspension, cell suspension culture or suspension medium. Now, in this case, in our case, actually, this is a overall work plan of the in vitro study. You can proceed in 3 different directions. One is straightway you can take the explant and then direct organogenesis. The leaf tissue is very responsive. You know, Solanacea is a very responsive plant. It's an ideal plant for class material or research work, because you don't have to face any problem while regeneration of this particular one. Easily they can regenerate. So here actually the plants have been regenerated through leaf culture, organ culture system, and you can get shoot buds later on, shoot elongation, and finally the plants, and they have been successfully hardened and transferred into the field condition. In another route, actually, you can go for callus induction, where you have to proceed either shoot organogenesis or embryogenesis. In our case, we have just taken up the shoot organogenesis part, or we didn't try for embryogenesis because that was not necessary. We get profuse shoot organogenesis through this particular system, and then you are actually coming to the same path. And in a third system where shoot tip culture can be used, where there is no callus phase, you know, so that to get the genetically stable clones, this is the best system, and this is the. So we have tried in 3 different directions for in vitro propagation of plants, and we get success in all these 3 steps as well. Now, this is just a slide we can skip. What are the things required for in vitro propagation? That means the methodology part. Actually, just I will tell you that for callus induction, actually the plant growth regulators are actually mentioned here. You have for callus induction, you need a set of plant growth regulator, set of hormone like auxins mainly, and then for shoot regeneration, actually here the cytokinins play a very important role, and we have tried different cytokinins and to see the efficiency of regeneration that we will be presenting in a moment. And then rooting is actually achieved by either Na or IBA, both are auxins, and we got good result with that also. So this we have achieved with direct and indirect organogenesis and plant regeneration, and followed by their assessment of genetics fidelity through molecular markers. Now, just to show you the callus induction part, the preliminary part, actually you have tried auxin Na and combination of auxin and cytokinin. You can see here that callus is primarily induced with auxin. More or higher amount of callus is induced with Na, also with BAP to some extent. Now in the photograph also you can see that the upper panel actually giving your friable callus, which is induced only by Na. The lower panel actually the combined result of your IA and Na and BAP, that is auxin cytokinin combination. You can see the in this case DEF here the callus is more compact compared to the friable one, the upper ones. So far as the plant regeneration is concerned from the callus through organogenesis, It is from callus tissue. You can see that the plant regeneration has been achieved in different cytokinins, excepting the kinetin. Kinetin is incapable of generating any shoot organogenesis. The best result was actually obtained in TDZ in low concentration, and kinetin and other, and to some extent 2-IPA also is giving some good results. So the photograph also you can see the ABC, this is TDZ, this is IPA, and this is BAP. More or less the general health of the shoots and plants are good, although the number is actually higher in case of TDZ. The lower one is also the same. This is just to show you how the shoots are actually regenerated from callus tissue. This is the case of leaf explant. Direct, I think there is. Here you see, in contrast to the previous one, here again plant regeneration is actually shown, but here interestingly, the better result is obtained with the benzyl aminopurine, not TDZ. The earlier one was TDZ was the best hormone, best cytokinin for shoot regeneration. Here it is better with the BAP at particular concentration, and to some extent 2-IPA is giving rise to also shoot regeneration from leaf directly without any callus. So these are the photographs. You can see that leaf is giving rise to stepwise, it is actually getting multiplied, and finally you are getting the shoot regeneration. And this is the shoot tip culture, just to compare the three different roots. Here, the shoot tip culture, there is no callus formation at all, and you can see that 2-IPA is giving the best result for direct shoot tip multiplication and shoot tip culture along with BAP. So we just checked the 4 different cytokinins and we got results, at least positive results with all those 4, excepting a few cases. But for a particular type of organogenesis, one particular type of cytokinin is giving best result. This is the interesting part to be noted here. This is just to give you the overview of the different cytokinins, how they are actually responding to different type of cultures. So that this is not very clearly visible, of course. Anyway, the red one is a direct shoot regeneration, the green one is a shoot tip culture, and the blue one is a callus mediated. You can see you are getting positive response in almost all the cases. This is just to tell you the root induction. Root induction was achieved in basal medium as well as NA and IBA, but the interesting part is that in most of the cases, people who are actually working with the plant tissue culture in this particular plant tissue culture area, we have seen that in many cases, the root induction is there. For some plant, root induction is not at all a problem, but for some other plants, it is a really difficult task, particularly for tree species. We have faced tremendous problem in root induction in tree species. So here actually root induction is not that difficult, but the problem is that once you have root induction, the root is converted into callus. And that is a very inconvenient part of our work, because once you are trying for field transfer, subsequently, the plants do not survive because of the callus mass present at the base of the root. So we always try to get direct roots without any callus phase, and the basal medium has given more or less good result, and we picked up this particular part. So normally we do not require to add any auxin for root induction. Simply, if we omit the cytokinin from the shoots, the rooting actually takes place. This is the last part, that is the how the plants are actually transferred to the field condition, and we have actually we did it through stepwise acclimatization process. That's a standard process developed in every lab. So we have also followed the same thing. So the plants are actually gradually taken from lab to land condition, not directly jumping to the field, to the soil, but it is actually processed through stepwise acclimatization process. And finally, they are actually brought to the field condition, and the success rate is more or less good. 70 to 80% plants survive, and they are established in the soil condition. So this is the first part. It's a simple thing, and we did not face much problem in having micropropagation of this plant, and the number of plants can be very, very high, and which would be subsequently utilized for other purposes. So then finally, we actually checked the genetic fidelity of the plants generated through different pathways, including the callus-mediated pathway, callus-mediated route. And in all the cases, actually, we have seen that the plants are perfectly stable genetically. So here you can see we have actually performed RAPD analysis, and here we have attempted with the 10 primers, RAPD primers, and out of those 10, actually 6 gave consistent result with monomorphic banding pattern. Here you can see the M is the mother plant in each case. These are all different primers, RAPD primers. This is the ladder, and you see the M is the mother plant, and 1, 2, 3, 4, 5 are actually plants regenerated in 5 consecutive generations. So that means we wanted to see whether this particular genetic stability is maintained in culture with age, or it is actually changing as the culture is actually growing older. So we have found that till 5th generation, that means 10 months, we actually tried for 10 months, and then we saw that plants are actually giving them monomorphic bands. That means they are quite genetically stable. They are actually consistent with their mother plants. So this is actually the last part of this. And then coming to the second part, that is secondary metabolite studies from in vivo and vitro state tissues. Here, actually, our objective is to two objectives we have. First, in the first part, we just tried to check which particular plant organ gives the highest amount of secondary metabolite, total secondary metabolite, or different fractions. And once you identify that particular organ, we will initiate culture, and then from cultures, actually, we will again see whether the cultured tissue is giving in normal condition, giving higher amount of secondary metabolite compared to the field plant, or we need to elicit all those cultures in order to get higher amount of secondary metabolites. So that is the more or less the objective of this particular part. And this is just secondary metabolite. I did not give the definition of secondary metabolite. All of you know. It is actually a phytochemical, and the metabolites which are actually produced by the plants, but they are not directly utilized for the growth and metabolism of that particular plant itself, plant or organism, whatever you say. This is that methodology. I can skip that one. We have actually estimated different kinds of secondary metabolites, different groups. Actually, standard methods are available. Estimation of total alkaloid by drag and drops alkaloid estimation method. Then estimation of total phenolic contents by Folin-Cl2 method of singleton. Then estimation of flavonoid content by aluminum chloride calorimetric method. And then estimation of total tannin content by Folin-Cl2 method. And then finally, the solasodine, this is a single fraction out of those alkaloids, steroidal alkaloid group actually. And we need to utilize the HPLC part, HPLC instrument for assessing the solasodine against the standard solasodine. Now, regarding the quantitative estimation of total phytochemical contents, it has been found that the leaf tissue, in the field-grown leaf tissues are actually giving the highest amount of almost all the four types of secondary metabolites, like phenolic content, flavonoid, tannin, and alkaloids, etc. So leaf tissue is actually the best performing tissue, followed by root and the other organs. Normally, we had an idea that the fruit is actually rich in the ripe and unripe fruits are actually rich in secondary metabolites in solanaceous members. But in this case, we found that leaf is actually having the higher amount of secondary metabolites. And the total phytochemical contents in vivo and in vitro tissue, this particular figure will give you the phenolic content, the flavonoid content, tannin content, and alkaloid content. And you can see that the leaf tissue of field-grown plant, and here it is the callus tissue. Callus is always the less producing tissue. Compared to that, we have the leaf. These are the results of these particular bars are actually representing the amount of phenolic contents in the leaf of regenerated plants, in vitro regenerated plants in presence of different. In fact, I want to highlight here that BAP, kinetin, 2-IPA, and TDZ actually have been compared for the production of phenolic contents, phenolic compounds in the in vitro plant in vitro system, and we found that the 2-IPA is giving the maximum amount of phenolics in the cultured leaf tissues. Similarly, regarding the total flavonoid content, it is the kinetin which is actually giving the best result in the regenerated plant leaves of the regenerated plants or tissue culture plants. And in case of tannins, actually, TDZ, in fact, TDZ, 2-IPA, and kinetin, they are all good performing, but TDZ is slightly better here, and it is giving the regenerated plants, the leaves of the regenerated plants are giving the higher amount of total tannin content. And regarding the alkaloid content, it is the 2-IPA, which is giving you the better result. So just to give you an idea that the cytokinins, different cytokinins are actually responsible for differential synthesis of different phytochemicals or secondary metabolites in the cultured tissue. This actually we got here. And regarding the solasodin content, which was actually measured through HPLC method, it is found that there is a drastic or dramatic increase in the solasodin content in the leaves of in vitro plants in presence of 2-IPA, which is almost 46.78 milligram per gram of tissue, compared to the in vitro leaf, the field-grown leaf, which is only 12. So BAP and 2-IPA, both are good performing one in this case. So this is a very good result. We should say that the solasodin content is drastically increased in presence of one particular cytokinin in cultured tissue. Now we are coming to the next part. So in the first part, actually, we have seen that once we initiate culture, once we regenerate plants, in presence of various hormones, actually, what are the performances? What are the secondary metabolites produced? In the follow-up part, actually, here we have actually experimented on the known some growth retardants, which are commonly used for different purposes in the tissue culture or plant biotechnology experiments. And those are actually chlorochlorine chloride, CCC, and simidol and paclobutazole. We selected these three plant growth retardants, and these are all synthetic compounds which are used to suppress the growth rate of the plants in a desired manner without lowering the plant productivity and vigor. In fact, we have been working on this particular field for quite some time for some other purpose in our laboratory. That is, we are actually working for in vitro storage of plants and tissues for a reasonable length of time, that is 1 to 2 years. We are we are actually working on that. So that is called in vitro conservation or in vitro storage of culture tissue, which can, this is a particular program of genetic conservation of plants and here. So in orchids, actually, we got very good results. So there also we have used this kind of what is called these plant growth regulators for suppressing the growth of the cultures. Because once you are actually trying to reduce the subculturing, to increase the subculturing period from say 2 months, 8 weeks to say 10 months or 12 months, actually what we have to do, we have to add some compound in the culture medium which will suppress the growth of the cultures but without damaging the general health. So these are very common ones actually you use. We have some other ones. So here actually we have used these plant growth retardants for some other purpose. That is, if we can suppress the growth of the cultures, it has been observed, like in other plants, that the secondary metabolite content is gradually increased. This is a general concept, general observation in other plant groups. So we have taken up that thing and we use these three in our Solanum erianthum cultures, and we'll see the results. This is just a slide where we have mentioned these things. Now, regarding the callus cultures, actually you can see that we have used CCC, ensimidol, and paclobutazole in the callus tissue. And we can see that CCC actually is giving the highest amount of phenolic content or higher amount of phenolic content compared to the the rest of the plant growth regulators, plant growth retardants. And this is your callus, that is the control. And this is also callus, but it is in presence of CCC, it is greatly increased. From 6 to, you can see, almost doubled. Now, total flavonoid content also, CCC is giving better result compared to ensimidol and paclobutazole. Paclobutazole at 1 milligram, actually it is giving more or less good result, but CCC is always better. Regarding the total alkaloid content, again, CCC is giving 0.5 and 1, both are actually 1 milligram per liter, both are giving good result. And only the tannin content is actually giving some different result, where paclobutazole is another actually gibberellic acid inhibitor. This one is giving better result for your secondary metabolite production. So here again, you can see that different types of growth retardants are actually working differently on different phytochemicals or different secondary metabolites. So this was our result here. So previous one was in callus tissue. The subsequent one is actually on the leaf tissue of the regenerative plants. Here also phenolic content is better or greater in CCC, and flavonoid content is greater or better in paclobutazole. Tannin content is better in ensimidol. This is the only difference, because ensimidol is not giving so much better result compared to the others, but here it is giving a good one. And it is a control here. You can see that control is also a good performing one. Tannin is also present in higher amount in the control tissue. That is the normal without normal cultured plants or regenerated plants without any plant growth regulators, plant growth retardants. Total alkaloid content is also very high in paclobutazole treated cultures. So these are the overall findings. The content of the secondary metabolites usually showed an increasing trend in cultured tissues treated with various plant growth retardants, such as chloroquine chloride, and ensimidol, and paclobutazole. So this is a positive result, that if you add plant growth retardants in the culture, if you can suppress the growth of the cultures, so you get lesser amount of biomass, but your secondary metabolite content is actually increased. So for actual a better recommendation would be that this kind of work should be subdivided into two parts. In the first part, you just grow as much as possible. You grow and regenerate plants in very high amount to get a higher biomass, and then transfer or change the media condition, add these particular plant growth retardants in your culture, so that the cultures, instead of growth, they will be now directed towards the synthesis of secondary metabolites. So you can get the maximum benefit from this kind of two-step culture procedure. Now, the total phenolic content is highest in tissues treated with CCC. The flavonoid content was high in paclobutazole treated tissue, and the total alkaloid content was increased markedly by paclobutazole treated tissues. So that means, again, you can see that different plant growth retardants actually are acting differently on the production of different kinds of secondary metabolites in cultures. And in the last part, actually, we have attempted to raise hairy root culture of this Solanum erianthum, and then to see whether the different rhizoclones generated in vitro are performing better or not compared to the other previous ones. Now, hairy root culture, we have already learned from Professor Saksena's lecture that it is a type of culture produced after the infection of explants by the gram-negative bacterium, it's a soil-borne bacterium, Agrobacterium rhizogenes. It produces hairy root, and hairy roots can, if you see the roots of the field-grown rice plants. So from that, actually, concept came that instead of taproot and branch root, secondary root, it is the tuft of hairy roots actually generated from a particular spot, particular point. So this process takes the advantage of naturally occurring hairy root disease in dicotyledons, mostly. And the importance of hairy root is the most important is autotrophy. Like your crown gall, there also the gall is formed, the tumor is formed without having any without application of any plant growth regulators or auxin or cytokinin. It can generate the tumor tissue. Similarly, here the hairy roots are generated without the help of any plant hormones, and it has very fast and rapid growth capacity, these hairy roots. That is another importance or advantage, and high level of secondary metabolite production by the hairy root. This is a known fact. In majority of the cases, the hairy roots are capable of generating higher or accumulating higher amount of secondary metabolites in different types of plants. So this is just to give you the glimpse of the Agrobacterium rhizogenes. Like your TI plasmid, it has also got a RI plasmid, which contains a lot of genes and the border sequences, left and right border sequences, and then the virulence genes, etc. Everything is there. So it is also capable of transferring the tDNA of the RI plasmid, and that tDNA is carrying the genes which are responsible for generation of the hairy roots. So that is transferred almost following the similar type of mechanism. And here also, it is capable of infecting a wide range of plant species. The bacterium harbors RI plasmid, which causes the formation of hairy roots upon infection. These hairy roots, after induction, continue to grow in vitro without supplementation of any plant growth regulator in the culture medium, mainly auxin. So auxin is actually not necessary. This is an infection procedure. Everybody knows it. This is just a textbook knowledge that RI plasmid, this is tDNA, and these are the proteins generated by the Vir gene complex, and the virulence proteins are all It is the T strand, which is coated with all those proteins, and it is penetrating. This is the transport channel penetrating the host cell and ultimately getting integrated into the host genome, and it is getting expressed, and the hairy root is induced. This is our work plan. The culture maintenance of Agrobacterium rhizogenes A4. This particular strain was actually supplied by Professor Suchitra Banerjee. We didn't have this in our department, so I had to wait for some time anyway. And then the leaf explant. So the culture is maintained, and the culture is actually the leaf explant, which is actually taken from our in vitro culture system. That infection of explant by Agrobacterium rhizogenes, this is all known fact. I'm just telling you. And then co-cultivation of the explants with Agrobacterium rhizogenes cultures. So you take the explant, that is normally the leaf disc of the culture grown plants, regenerated plants, and then the bacterium itself, they are actually all co-cultivated for certain time. The detailed methodology I'm not going. And then establishment and maintenance of hairy root culture, those are called rhizoclones. And then confirmation of the tDNA localization in rhizoclone by PCR amplification using primers of role A and role B genes. This is a standard method that the genes which are actually located within the tDNA portion of the RI plasmid are actually, the primers are prepared from that for that role A and role B gene, and they are actually checked through PCR, and this is the detailed methodology. So they are checked through PCR so that whether that particular role A and role B gene are actually integrated into the host plant or not. I mean, that means in the rhizoclone, the roots which are actually generated, the hairy roots, whether the cells of the roots are actually having those role A and role B genes or not. If the presence of these two genes are actually detected within the cells of the host plant or host tissue, then you are sure that the transformation process is successful, and then you can wait for the root induction, hairy root induction, you can follow up that. So this is one. Cephalotoxin is used to eliminate the extra bacterium. This has been done in our laboratory successfully, and we succeeded in getting. And in the next one, you can see here, the first one A is here. This is the control. In fact, actually there is no infection here. There is no infection actually done here. See how it is done actually. Co-culture, before co-culturing, you have to prick the leaf tissue, pricking with some needle or something like that along the midrib portion, and then put that leaf disc or leaf segment in a system, in an aseptic system where you add the particular bacterium. So this Agrobacterium rhizogenes bacterium will now enter the cell through those injured portions, and they will be ultimately getting integrated. And then we have to wait for the induction of the roots. So in the B slide or B photograph, actually you can see the initiation of hairy root, although it's a very small one here. Arrow is on the different direction. Anyway, but we can see the root here. Then C is gradually, it is increasing. Root elongation in semi-solid medium. D is further growth. E and F are full-grown hairy roots. And here actually there is no auxin added. So it is auxin-free medium we are actually growing these particular hairy roots. Now these hairy roots, actually C, E and F are actually two rhizoclones I have given. Actually, we have got some more, but we have compared these two for their performance. You can see here, this is just to show you how the hairy roots are characterized through your molecular technique, and to see whether the tissues are actually having that particular tDNA portion, or whether they are transformed or not. Actually, this is the particular protocol methodology. And then this role A and role B, these two genes have been tried, and you can see their PCR condition is more or less same. Only small difference is there that the annealing temperature with the primer is slightly different in case of Role and Role B gene, 55 and 57 degrees Celsius. And the elongation, in case of chain elongation, actually, the slight difference is there regarding the time schedule. The rest of the things are all same. And here you can see the expression. This L is actually the 10 kb DNA ladder, and then PC is your positive control. That is the plasmid itself, and N is non-transformed root. We have to always take the non-transformed root, non-infected root that does not contain any such Role and Role B gene. So it is actually blank. And then 1 to 5 are, these are the rhizoclones, HR A, B, C, D, E, like that. So different rhizoclones we have presented here. And here, the PCR products are expressed or represented in relative abundance of Role and Role B in rhizoclones with. So this is just the difference in the intensity. Actually, the relative abundance of that particular gene actually are different in different rhizoclones. So that is just to show you, but in all the cases, actually, the roots are transformed. And the growth, this is the growth of the rhizoclones here. You can see that the blue line is actually the non-transformed root. So there is not much growth without any added auxin in the medium. But these are actually showing very high growth rate with all these are all transformed roots A, B, C, D, E, like that. So this proves that there is a difference between the non-transformed and transformed roots so far as the growth rate is concerned. Now their phytochemical background, that means again the total phenolic content, flavonoid content, tannin content, and alkali content we have checked. And this is the field-grown leaf. This is the root, and here roots. Again, the content, the phenolic content is much higher compared to the non-transformed tissues. Similarly, total flavonoid content is quite high in both the rhizoclones. Tannin content is also giving a good boost here, and then total alkaloid content is also good. So it proves that following the previous works on different plant species, here also in Solanum erianthum, we are getting very high amount of secondary metabolite production in the hairy root condition. This is solasodin content. Similarly, solasodin content is also increased. The study showed that the solasodin content of the rhizoclone was 16.29 here, whereas that of the leaf tissue, normal leaf tissue or cultured leaf tissue is much less, 12 point something. So the result indicates that 1.32 fold increase in solasodin content in selected rhizoclone compared to the leaf, that is remarkably high. Quite high, not remarkably high, but it is quite high. Now, finally, the conclusion of this particular presentation: an efficient in vitro protocol for rapid clonal propagation of Solanum erianthum in both callus-mediated and direct organogenesis pathways has been established. The genetic fidelity of the in vitro regenerated plantlets via callus-mediated organogenesis has been assessed using standard molecular techniques. The in vitro generated well-rooted plantlets have been established to the field condition following standard hardening protocol. The manipulation of media components has been a strategy for improvement of secondary metabolite production by Solanum erianthum cultures. Solasodin content increased significantly in presence of cytokinins. Stable hairy root cultures have been established in Solanum erianthum by infection of Agrobacterium rhizogenes A4 strain and specific rhizoclones. And specific rhizoclones have been raised with high growth rate and increased solasodin content. So that means you can always progress stepwise, starting from cultural condition to finally the hairy root systems, because it is an established fact that normally the cultured tissues, secondary metabolite content is decreased drastically. So that was a known fact. So from that, actually, you have to elevate your work in up to a certain level, that whether the secondary metabolite content is actually augmented reasonably so that it is comparable to the field condition, plants of the field condition, so that it can be utilized for commercial purposes or commercial purposes or something like that. So in this case, actually, this plant, actually, we have established a particular method or protocol that this plant now can be utilized, because this is one of the highest solacodin containing plants of the Solanum genus, along with your Solanum cassianum and some other plants. So I think that's all for today. Thank you.
Thank you, sir, for giving a very lucid and scientific lecture. You deal the subject from the field condition to the scientific validation with different techniques. So it is very knowledgeable, and we are enriched with your lecture. So you talk about how many species of this particular Solanaceae family consists of. There are around 2,300 species that you have talked about, and what is its medicinal uses you talk about. And because of the medicinal uses that we have at the background on that, you selected the plant and you go for in vitro culture, and different in vitro culture system were conducted, and found out that all the tissue culture that you experimented was found to be 80 to 70% when you transfer into the field condition. And now the secondary metabolite system, how you can maximize the secondary metabolites in the plant through different process that you have discussed in detail. And we are very much enriched with the stepwise explanation in detail. So it is a very knowledgeable and it is a very informative lecture that we have. What I wanted to emphasize here is that you use secondary in vitro culture. And in vitro culture, why we use in vitro culture? Because we wanted to maximize the secondary metabolites through different culture system. And that was very interesting. And genetic fidelity, you talk about how the regenerating plants changes in its genetic composition if we use this and that chemicals for the growth, and that you have checked for all the different parts of the plant, and that is found to be very stable. And at the same time, you use suppressant retardants growth of the plant. Why we use this? Because we wanted to suppress the other parts of the plant that will interfere with the production of secondary metabolites. And you have discussed in detail the different retardants and that retardants in different parts. And so that part also you have explained very lucidly, and you have shown us that the growth retardant enhance the secondary metabolites in different parts of the plant. Not only that, you go to non-interference of growth hormone in production using agrobacterium. You have discussed about that, and that is one of the way how we can enhance the secondary metabolites without using growth hormone or synthetic chemicals. So in that also you have shown that there is definitely a very good secondary metabolite production using the agrobacterium. So in all together, you give us a very informative knowledge on the subject. So thank you very much for your lucid presentation, sir. And we are glad to have you again in our midst. And thank you for the participant for patient hearing. And if there is any question, one or some of you can put up some question to our speaker, Professor Nirmal Banerjee. Thank you. Any questions from the audience? At least one or two questions.
নির্মল দা
বলো।
বলছি যে the variation that we get out of tissue culture or the genetic change in the constitution, I mean, whatever the change we are getting during tissue culture, so is due to a mutation. Should we call it mutation? সেইটা আমি
genetic change যেটা হয় এর ক্ষেত্রে আমাদের হয়নি। In our case we didn't detect any genetic variation, but when genetic variation occurs, it occurs. Yesterday I was talking to I was actually discussing with another lecture of some girl, I think in some other session. সেখানে যেটা হচ্ছে যে once you have callus induction, callus is an abnormal tissue, and that abnormal tissue contains lot of abnormal cells along with some normal cells. সুতরাং যে সেলগুলো abnormal cells আছে, those cells are not normally capable of regenerating plants. The normal cells in that particular mass actually will give rise to your plants. যদি কখনো কোনো abnormal cells বা abnormal plant তৈরি হয়, হতে পারে, there is a chance. In that case, actually, those plants are actually genetically variable plant, we should say, and those variability, তোমার যেটা প্রশ্ন সেটা whether that particular variation is actually retained in the field condition, or it is maintained in the field condition, plants of the field condition. সেইটা so এটা কিন্তু কখনো কখনো আমরা তো RAPD ISSR মলিকুলার মার্কার দিয়ে চেক করে বলে দিচ্ছি, কিন্তু যতক্ষণ না সেই প্লান্টটা at least 2 to 3 generations tested হবে in the field condition, we cannot release that plant as a variant. We cannot prescribe that plant as a genetically variable plant. মনে করো তুমি say you are heading towards বা তুমি খুঁজছো হয়তো যে Phytophthora resistant একটা potato plant, lead blight of potato disease resistant plant you are actually generating through your culture through this kind of genetic variation. সেক্ষেত্রে তোমাকে কিন্তু প্লান্টটা রিজেনারেট করে তোমাকে 2-3 জেনারেশন দেখে if that particular resistance character is actually retained by the plant, then only you can release it in the market. Otherwise you should not do that. বুঝতে পারছো? সেই জন্য অনেক সময় অনেক চেঞ্জ গুলো হয়, সেগুলো আবার যখন আমি ফিল্ড কন্ডিশনে আসছি তখন সেগুলো ফিরে আসে, it comes back. So that is actually, those are not mutation, those are actually epigenetic changes occurring during culture in the form
and one more thing, maybe it is not related to your work, the epigenetic change or the genetic change or the change in the nucleotide sequence. Okay, and whatever happening at molecular level, so is it random or non-random event?
এটা obviously random event.
বলছি কেন, কারণ ন্যাচারাল এভলিউশন মানে আমরা জানতাম যে মিউটেশন ইজ র্যান্ডম. I came one paper yesterday, it published in Nature, it says that mutation is non-random.
Mutation is non-random মানে তোমার যেগুলো আর্টিফিশিয়ালি করা হয়, যেগুলোকে তুমি বলছো যে সাইট ডাইরেক্টেড Phytogenesis, যেগুলো আছে, those are actually different aspects. কিন্তু নরমালি মিউটেশন গুলো র্যান্ডমই হয়, কোনো we don't have any control over mutation. That is the thing, unless it is artificially done, like your site specific Phytogenesis ইত্যাদি ইত্যাদি. কিন্তু আমার যেখানে এখানে এটা বলা রয়েছে সেটা হচ্ছে যে it may not be always in the DNA sequence changes in the DNA sequence part. এরকম হতে পারে যে কোনো একটা one particular gene was expressing in the normal condition, And due to culture process, actually certain repressor or that particular some kind of repression of that gene is actually taking place for certain time or for a particular period of time, so that that particular gene is repressed and you get a changed character. And in the field condition, when the plants are actually brought back to the field condition, that repression is removed and the gene is actually started functioning in a normal way. সুতরাং আবার পাল্টে যাচ্ছে। এরকম কেসে কিন্তু হতে পারে। Instead of going directly into the DNA sequence, changes in the DNA sequence in the form of mutation, some repressor protein এর মধ্যেও কিন্তু এখানে মানে তাদের রিপ্রেশন, তাদের ডিরিপ্রেশন হতেই পারে। সুতরাং এটা কিন্তু আরো ডিটেইল স্টাডি না করলে কিন্তু বলা যাবে না। সব রকম রিপোর্টই আছে। There are changes, permanent changes also. Large number of disease-resistant plants have been released through this tissue culture system, which are actually established in the market. মানে bypassing the genetic engineering part. বুঝতে পারলে তো? এটা একদম প্যারালাল একটা জেনেটিক ইঞ্জিনিয়ারিং করে you can create change, you can create a particular new variety without genetic engineering, which is a controversial part nowadays. You can use this particular in vitro system, tissue culture system, to generate some kind of resistance. Okay.
Thank you, Nirmal Lada.
Okay, thank you.
Anybody? Acha, sir, if others are not asking, I just want one question. Sir, for the induction of agrobacterium to the cell so that we can get maximum secondary metabolites. So in the process, why we choose leaf tissue for the induction purposes and why not real root cells? What could be the reason?
Yeah, I don't say that it is not possible. You can always take any living plant tissue, any living part of the plant. You can take internodal segment, you can take root segment and try to do that. Thing is that the roots, we can also grow roots in the cultural condition, in the tissue culture, we have seen. For regeneration of plants, actually, the final phase is the root induction. Okay, so that is one thing, that is one part. But here, what is happening, certain host cells are getting transformed by the introduction of the tDNA of the RI plasmid. And because of that incorporation of that integration of that particular tDNA portion in the host genome, it is coming from a prokaryotic system, you have to remember. But it is getting integrated and the particular genes which are being carried by that particular tDNA of RI plasmid actually getting expressed if they get the congenial environment within the host cell. Once they get the congenial environment for expression of those genes, they will start producing the hairy roots. Now those hairy roots it is in fact a kind of disease, plant disease, like your crown gall. But this particular phenomenon has been utilized by the plant biotechnologists for production of secondary metabolite.
Okay, thank you very much, sir, for your lucid explanation. And if there is no more question, then I will hand over the time to the anchor. Take your time. Thank you.
Thank you very much, Nirmal Lada. Thank you very much. আমরা সবাই মানে very happy ki apni join করলেন. So we are very happy that we listened today again after a gap. আবার একটা গ্যাপের পরে সব স্টুডেন্টসরা সবাই আমরা আপনাকে শুনতে পেলাম। তো this is very good. I request Rukhsana Irani, our organizing secretary from Raidighi College. So please share the token of appreciation of this session. Dr. Gauri Saxena, Professor Rita Kundu, and Professor Nirmal Banerjee. So please.
Dr. Roy, am I properly audible? Am I audible?
Yes, you are audible. Yes.
Okay, good afternoon, everyone. Thank you so much for giving me this opportunity that I'll be showing the screen for the certificates on behalf of the organizers. So I'm just sharing the screen. Yes. First of all, I would like to present this certificate for Professor Gauri Saxena, Department of Botany, University of Lucknow. Her lecture was tremendously beneficial for the students. Thanks, ma'am, for being with us today. I'll just zoom it a bit. Okay. Now the next certificate. Professor Rita Kundu, ma'am, Department of Botany, University of Calcutta. We are extremely thankful to you, ma'am, for your lucid and informative speech. We were really enriched by your lecture.
Thank you, Professor Roxana.
Our organizers are extremely honored to present this certificate to Sir Professor Nirmal Banerjee, Department of Botany, Visva-Bharati University. The lecture and the way he presented the entire matter, really for researchers, it is something insightful and thought-provoking. Thank you so much, sir. It was really an experience that we will be cherished forever. Thank you, sir.
Thank you.
So this is for now. Now it's over to you, Dr. Roy.
Thank you, Ruxana. Thank you very much. Now, Devoshmita, please, we will assemble at again at 2:45 for the valedictory section. Now I'm hand over to Devoshmita to conclude the session.
Thank you, ma'am. Thank you, sir, for the lecture, and thank you, ma'am, Ruxana, ma'am. So after lunch, we will meet again at the valedictory session at 3 p.m. So please join the same link are you in. So soon. Thank you all.
Okay, thank you.
Thank you. Thank you.
Thank you, Angelika and others.
Thank you, Rita Di. Thank you. Abar validity session ma'am join korben. I request all to join in the validity session, please.
Okay.
Okay, Angelika. See you.
Thank you, sir. Thank you.